Great.
Everyone, and welcome to the first Black Queer Town Hall in STEM. We are so excited to have you all here for you to hear some phenomenal science from our speakers today. Ryan and I created this event to mirror the Black Queer Town Hall that was done this past summer by Bob the Drag Queen and Miss Peppermint to celebrate experience and excellence by cultivating a community, sharing knowledge and uplifting voices and culture. We wanted to do the same thing within the STEM community and increase the visibility of Black queer scientists to make STEM more equitable and safe for everyone to be out. Thank you again for attending, and we hope you have a fantastic time. And don't forget to ask questions, because that is the backbone of science discovery.
Speaker of the day is Dr. Avery Posey. He received his PhD in genetics from the University of Chicago. Subsequently, he then went on to do a postdoctoral fellowship in Dr. Carl June's lab, which pioneered CAR T-cell immunotherapy to treat cancer, and is now an assistant professor in the Department of Systems Pharmacology, Translational Therapeutics at the University of Pennsylvania. He has also been an active voice at the University of Pennsylvania to ensure that the Black Lives Matter movement is more than a moment. Please welcome Dr. Avery Posey.
Hello, my name is Avery Posey, and I'm an assistant professor in the Department of Systems Pharmacology and Translational Therapeutics at the University of Pennsylvania Perelman School of Medicine. Since I'm the first speaker of this morning's presentations, I've been tasked with introducing the topic of intersectionality. I used to think that topics of identity and politics were meant for home and not for professional events. But I think the entirety of 2020 has demonstrated that this could no longer be the case and should no longer be the case. Before I begin, I want to share with you that I'm pre-recording this on the evening of January 7th, 2021, one day after we witnessed an insurrectionist mob of white supremacists storm the US Capitol to overturn the will of the American people and ensure that another white supremacist remains president for another 4 years. This action, as well as the many failures in public health in 2020, combined with the atrocities and injustices committed against marginalized citizens and the president's backing of white supremacists as very fine people, should shake us all. When we are this fractured as a country and as a people, we cannot maintain the sanctity of peace, the bliss of ignoring identity in the workplace, the leisure and joy with which we may have lived our lives pre-pandemic. When we are this fractured, everything breaks down. And so I encourage everyone to step outside of their comfort zones, to embrace the differences in others as additives to your own lives, and to strive to understand how people who may not look, live, or love like you carry out their lives. I have some upfronts to share before I begin to discuss my view on this topic. First, I am not a scholar of race, sexuality, or gender. There are actual experts in these topics who have dedicated their careers to the study of intersectionality. If you are interested in more detailed study of these topics, I encourage you to seek out those experts. Second, the comments I will make are mine alone and are not endorsed by any organization or institution, nor have I asked for that endorsement. So intersectionality is defined as the interconnected nature of social categorizations such as race, class, gender, and sexuality as they apply to a given individual or group, regarded as creating overlapping and interdependent systems of discrimination or disadvantage, For as long as I can remember, I've been Black. As I'm sure we are all now very aware, Black people face the major obstacle of racism, have led the civil rights movement, which has greatly benefited the lives of Black people, and also have a uniqueness in their culture, their language, music, and arts. Also, for as long as I can remember, and I think I was in the second grade when I first learned how to describe my feelings of otherness, I've been gay. Queer people similarly face an obstacle. Here, homophobia. And in the recent decade, marriage equality improved the lives of queer people. And similarly, queer people have a uniqueness in our culture, language, and arts. At the intersection of these two cultures are people like myself and others who will speak throughout this event, the Black and queer. individuals who exist within this intersection experience racism and homophobia, and even face homophobia from people of their own race and racism from people of their own sexual identity. Black and queer individuals also have a uniqueness in culture, language, music, and arts. Important for these groups and all marginalized and minority groups is a requirement for fellowship with individuals of their identity. This is one reason why Black gay prides are hosted in many cities annually, in addition to the larger gay prides. Spending time with people who share your identity can finally give you a chance to breathe and escape the feeling of otherness for a few days. Black and queer individuals have made major contributions to our society. Here I've highlighted Marsha P. Johnson, who likely would identify now as a trans woman, along with Sylvia Rivera. These individuals led the Stonewall riots that sparked the LGBTQ pride movement, which is over 50 years strong today. Bayard Rustin, pictured here along with Dr. Martin Luther King Jr., helped to organize the civil rights movement and the momentous 1963 March on Washington. Also pictured are famous authors like Langston Hughes, Zora Neale Hurston, and James Baldwin, and living legends like Alice Walker, author of The Color Purple, and Dr. Angela Davis, former Black Panther Party member who continues to fight for criminal justice reform so that men and women of color do not continue to be trapped in the prison industrial complex for minor offenses like possession of small quantities of drugs that citizens in several states can purchase legally today. And perhaps there could be more focus on jailing insurrectionists who desecrate and destroy our most significant and sacred government activities and institutions. While I mentioned that I had been Black and queer for my life, I did not come out until my first year of graduate school. And my desire to connect with more with my identity was fed by works from individuals who I used to read online then, but have now since become my friends, like Keith Boykin, who is the LGBTQ liaison in the Clinton White House, and E. Patrick Johnson, who is the Dean of Northwestern School of Communications. These individuals, through their work, dissected the world of politics and of normal life and centered their interpretations in their own identities as Black and queer men. Keith maintained a blog at keithboykin.com. It doesn't exist anymore today, but then it covered everyday political events from the last day. Keith wrote this blog every morning before 8 a.m. and I read it every morning before class. That wasn't an easy feat back then to cover the politics of the day, because back in 2005, I think we all paid less attention to politics. I doubt anyone knew the name of the Postmaster General in 2005, like I can rattle the name of Louis DeJoy today. The question that was posed to me for this talk was, why is the intersectionality of Blackness and queerness important for STEM? I wish I could tell you that Black queer magic was the only way we'll solve some major problem, that it's only us that can do it. That may be the case for HIV/AIDS, which surprisingly has not disappeared from our community and requires major attention. But identity doesn't mean that we're the only ones that can work on a particular topic. The answer, which I hope is clear by now, is that representation matters. This is why it brings joy to the hearts of Black queer individuals to see so many musicians in the current generations, like Tyler, the Creator, Big Freedia, Frank Ocean, and Lil Nas X. Stars who have all been awarded for their talents. It's also why I sought to read books like Elin Harris's Invisible Light trilogy that centered Black and queer protagonists, or why I would rush home from graduate school to watch the Logo series Noah's Ark with my friends. Seeing yourself in this media is important for validation and acceptance. Just as Black children today will no longer wonder if it's possible for a Black man to become president of the United States, for a Black woman of Jamaican and Indian descent to become vice president of the United States, for a Black pastor from Georgia to become one of 3 Black men in the US Senate and the first from his state. And its queer children would no longer wonder if it's possible for an openly gay man to serve in the cabinet of a US administration, or for a trans woman to lead a state's health department. This week's town hall will make it true that so too will Black and queer children who love to tinker in science, technology, engineering, and math no longer wonder if it's possible. It's possible. Thank you to the organizers for inviting me to speak at today's event, and thank you to the other participants for being an inspiration. Now, let's get to some science. The title of my talk is CARs for Solid Tumors, the sweet side of CAR T cells, reprogramming T cells to target glycopeptide epitopes. My lab is really interested in ways in which we can engineer the immune system in order to fight cancer. So we use a type of white blood cell called a T cell. A T cell, pictured here in this rainbow diagram in blue, is a lymphocyte. So it's one of the white blood cells of the adaptive immune system that can respond to an infection, like a virus, like coronavirus, or can also potentially respond to a cell that has lost its way, like a mutated tumor cell. And it does this through special equipment called the T cell receptor, or TCR. The TCR, along with a second signal called co-stimulatory molecules, when engaged, the TCR binds to MHC major histocompatibility class complexes, major histocompatibility complexes, like class 1 and class 2, that presents a peptide. When the TCR engages that MHC peptide complex and is co-stimulated, there's a signaling cascade within the T cell that in turn leads to transcription of some infectors, translation of that RNA into proteins like perforin, granzyme B, IL-2, interferon gamma, and the release of those molecules can lead to clearance of that infection, clearance of that tumor, and restoration to normality. TCR complexes with the peptide MHC complex, as I said, and co-stimulation provides signal 2. Both of these signals are required for a productive T cell response. The peptides that's presented here as AG in the MHC complex represents a snapshot of a cell's proteome. It's not the entire proteome, it's just a view of it. And our TCRs of our T cells are immense in diversity. So there's a diverse repertoire of TCRs that can virtually recognize any non-self peptide. That means that it's been tolerized to the cell proteome. Our T cells don't target our cells naturally. But the problem with cancer is that cancer arises from self. And so not every snapshot, every peptide that's presented in MHC is going to be that mutated protein that is leading to uncontrolled growth of the tumor. And so we need new strategies to overcome tolerance to tumors, because they arise from self, and direct the immune system towards tumor cells. And there are many types of T cell immunotherapies. We know that T cells can clear tumors because many of these types of therapeutics work and have removed large bulky tumors from patients. Many of these therapies exist, such as cytokine therapies, therapeutic vaccines, bispecific antibodies, checkpoint blockade is all the rage right now, anti-PD-1, anti-PD-L1, and anti-CTLA-4, because these molecules cut the brakes on the immune system and allows them to overcome tolerance that is to overcome the tolerance that's utilized by the tumor cells to shut down the immune response. And then at the bottom of the screen are 3 different types of adoptive cell therapies. These are T cells that have been removed from the body of a patient, grown to large numbers and then reinfused either alone or with genetic modification like CAR T cells or TCR engineered T cells. And the class of cells I'm going to talk about today are CAR T cells. So a CAR is a new molecule that is created, and it is introduced into the T cell with, in our case at Penn, a lentiviral vector. And the first type of CAR T cell that received FDA approval targeted CD19, a marker found on the surface of B cells. So this was actually a really great target for B cell leukemias and lymphomas. and actually some multiple myelomas. And so when we transduce these peripheral blood T cells of a patient, that new genetic material that the lentiviral vector delivers encodes for a chimeric antigen receptor, or CAR. That CAR allows the T cell now to recognize CD19 with this new molecule that's going to be expressed on the T cell surface. So when the T cells are reinfused into the patient, they now have the ability to see all CD19 positive B cells. And what happens is when they engage, the CAR is activated. The reason why the CAR is activated is because it contains signaling domains inside of the CAR that activate the T cell as well as provide co-stimulation. And so that leads the T cell to produce those effector molecules that I talked about earlier, like IL-2 and interferon gamma and perforin and granzyme B. And those are released and they kill the CD19 positive tumor cells. This has become a drug. It's now FDA approved. That drug is called tisagenlecleucel, Kymriah. There's another drug called, which brand name is Yescarta, and one other, Tecartus. So there are now 3 FDA-approved CAR T cells, all of which target CD19 for different indications. So specifically, what is the CAR? Well, the reason why those T cells are able to recognize CD19 is because the CAR is engineered with an antigen recognizing domain that is usually derived from an antibody. Antibodies are really great at binding to particular targets. I was going to say particular proteins, but you'll see later that antibodies actually have the ability to recognize multiple different macromolecules: protein, nucleic acid, lipids, and carbohydrates. And so you can engineer a chimeric antigen receptor to direct a T cell now not against a peptide MHC complex, but against something more tumor specific, if we were so fortunate as to identify an antibody that recognizes a tumor-specific antigen. The limitation of a chimeric antigen receptor is that because this new receptor is expressed on the surface of a T cell, this scFv must bind to an antigen also expressed on the surface of the opposing cell, the tumor cell. That is a limitation because what the natural TCR does is it can recognize a snapshot of a cell's proteome, both extracellular proteins and intracellular proteins. And so we lose the ability to recognize tumor-specific intracellular proteins by the use of a chimeric antigen receptor. We can only target tumor-specific extracellular proteins. However, there's also another limitation here, is that this is user-defined. Users, or scientists, define the antigen to which the T cell targets. So if that antibody recognizes an antigen that is not tumor specific, there is a potential cause of there's the potential to cause toxicity, in which the T cells will now attack a normal tissue or normal cells that are present in normal organs, normal tissue. And if that normal tissue is a vital organ, that poses some severe toxicity concerns. There are different generations of chimeric antigen receptors, here shown to your right. Ones in which the intracellular signaling is just that of this molecule, the CD3 zeta from the T cell receptor. And others in which they have been, in which the intracellular signaling domain of a costimulatory receptor, that second signal, has been added, either one costimulatory domain or multiple costimulatory domains in different generations. One benefit that a CAR has is that it does not require MHC. It does not require that MHC peptide. I've touched on this already, but that I need to drill it down a little bit more. That means that these T cells can work, these CARs can work in both CD4 T cells and CD8 T cells in the same manner. And so we don't need to select for CD4s versus CD8s, and that's not true of TCRs, which require a co-receptor, either CD4 or CD8 as the co-receptor for those particular molecules. And so I touched on this already, that the antigen binding ability of the chimeric antigen receptor is user-defined. And so in this first case, when the user defines the CAR to target CD19, these T cells can recognize and kill all CD19 expressing cells. So what I'm showing you to the right is actual clinical data from the first patient that was treated with these CAR T cells. So one day before infusion, you can see a snapshot of the patient's B cells. These are circulating, mostly leukemic cells in this individual that are all CD19 positive. 1 month after infusion, that CD19 positive, CD5 positive population that existed previously is gone and is completely gone. And that remains true in this individual for over 10 years now. And so in less than 1 month, CAR T cells had eradicated this patient's leukemia, and he has remained in remission for more than 10 years. That, I think, demonstrates the power of this type of technology. But how do we get this to solid tumors? And so that's where most of the cancer-related deaths exist. That's where we would like to have a major impact. If we can move this technology to, for instance, breast cancer, or ovarian cancer, or lung cancer, prostate cancer, pancreatic cancer, brain cancer, that's where we think we would have huge impacts in the survival of patients after they've been diagnosed with cancer. But there's a problem here, right? That user-defined antigen. How do we select for a tumor when we don't really have great cell surface tumor-specific markers? We would like to be able to only focus on the diseased portion of the forest, but not the surrounding healthy trees. How do we get there? And so the work that my lab has done is to focus almost exclusively on targets that are glycosylation related. So it's been known that differential glycosylation can generate cancer-specific or cancer-associated glycotargets. The class of glycans that we focus on are these truncated O-glycans. O-glycans are more simple than N-glycans. N-glycans are extremely complex, but O-glycans are a little more simple. But because of a change in O-glycosylation's machinery in tumor cells, there's a downregulation of O-glycosylation elongation, leading to a truncation that these residues make it to the surface of a cell when normally they would never be found as just TN antigen or just sil-TN antigen. And how do we actually get to that just TN antigen or just sil-TN antigen? Well, on every protein that gets glycosylated with an O-glycan, this happens on the amino acids serine or threonine. And the first step is that one of 20 different enzymes called GalNAc transferases, they add a GalNAc residue to that serine or threonine. And then that's somewhat sequence specific, some kind of polypeptide requirement there. And then there's one enzyme called C1-GalT1, which is also known as the T synthase, that it requires for proper folding an enzyme called cosmic to be present as well. And so that the expression of both of these enzymes then adds a galactose to that GalNAc residue. And that antigen is called the core-1 glycan or T antigen. However, commonly in tumors, downregulation of this machinery occurs, and one mechanism by which that happens is loss of cosmic, either a mutation in cosmic or epigenetic silencing of cosmic. It's also true that the same can happen for the C1-GalT1, the T synthase. And if either of that happens, a protein that would normally have an elongated O-glycan that would go further than this core-1 glycan is capped. It's truncated with just this residual GalNAc residue called the TN antigen. And there's another enzyme that will rapidly add a sialic acid to that TN antigen. We'll rapidly add that a sialic acid to that TN antigen, and that forms the sil-TN antigen. The presence of either of these at the cell surface is abnormal. And so that we call a truncated O-glycan. And it's been known for actually quite some time. This publication from George Springer in 1984 suggested that 80% of adenocarcinomas express the TN antigen. So that means that we've had the target for a while, but we really didn't know how to target it, how to best target it. and inter-CAR T cells. Collaborators of ours and friends and colleagues in Copenhagen at the Center for Glycosylation in Copenhagen also work in truncated O-glycans. And they've shown that if a cell were to lose cosmic, here they knocked out cosmic with zinc finger nucleases in an immortalized but non-malignant cell line. If a cell were to lose cosmic, it would naturally generate cell surface T antigen and also cell surface silyl-T antigen. Those cells would lose normal glycans like T antigen or silyl-T. And what happens functionally to those cells is that they would become more invasive in vitro as a skin line in vitro, but also more invasive in vivo. And in conjunction with that, they can lose some gene expression, such as loss of P53. And so that suggests that changing the O-glycosylation of a cell can induce oncogenic phenotypes. Right? There's signaling that occurs by loss of elongated O-glycans that leads to more oncogenesis. And relatedly, this has been demonstrated that increased levels of silyl-TN correlates with poor prognoses and certain tumor types. And so these are the type of tumor cells that we want to target. These are the type of tumor cells we want to get rid of. So how do we do that? Well, there are many types of heavily glycosylated proteins, and one that is well known is MUC1. MUC1 exists on the surface of many epithelial cells. It also exists on the surface of plasma cells, which is why multiple myeloma is of interest to us as well. MUC1 is found heavily glycosylated, where more than 50% of the molecular weight of the protein is just carbohydrates. O-glycans and N-glycans, here represented as N-glycans in red and O-glycans in blue. I apologize for the blurriness of this image. But in cancer, because of that truncation of the O-glycans, cancer-associated forms of MUC1 exist with short carbohydrates, such as TN antigen or silyl-TN antigen. And there are antibodies that have been developed that can recognize that silyl-TN antigen alone. The first one that we know of is B72.3. This is an antigen that's also known as TAG72. There are also antibodies that have been developed to recognize the TN antigen, such as 5F4. There's another called 1E3. And there are lectins that bind to these. Lectins are mostly plant-based proteins that bind to carbohydrates as well. There are human lectins as well, and mammalian lectins. And then there are antibodies like SM3, which doesn't bind to a glycan. It actually binds to a protein backbone that is now exposed by complete loss of carbohydrates. SM3 can recognize MUC1's protein backbone without any glycans, but it's also not disturbed if there was a TN antigen on this backbone. It is not care that the TN antigen is there. This antibody will still recognize this under-glycosylated MUC1, but it does not recognize the TN antigen. And then there are antibodies like 5E5, which we think is a novel class of antibodies that recognizes a glycopeptide. This antibody recognizes the combination of the TN antigen and the polypeptide backbone. If we remove either of those, the 5E5 does not bind. And we can show that here in this much clearer slide now, that 5E5 binds to MUC1 with the TN antigen and this ELISA at increasing amounts of peptide, increasing binding, and then it does not bind to MUC1 that does not have TN antigen. And interestingly, I will show you here that in this acylated oscillatory sheep mucin, so this is mucin that comes from a sheep that has been decylated, so it removed the sialic acid, so it presents with TN antigen. There is some binding. So that means that our antibody does have high specificity for TN antigen and has specificity for MUC1, but it also has some lower specificity for TN antigen on non-MUC1 proteins. So that is of interest, and it may be of value later to overcome the ability of a tumor to escape by losing MUC1 as long as those tumors retain TN antigen. And to the right, I'm showing you that in different tumor types, on these tumor microarrays like bladder cancer or breast cancer, ovarian, oral squamous cell carcinoma, pancreatic cancer, prostate cancer, kidney cancer, salivary gland cancer, and even multiple myeloma, we can identify TMF1 expression and bright TMF1 expression. And many of these tumors I highlighted with an asterisk. These are tumors that have low mutational burden. They're tumors that T cells would likely not target naturally. That even if these cells were, that even if we grew up large amounts of T cells from these patients and reinfused them, they likely would not target these patients' tumors because there are very few mutations to identify unless you find the one or few T cells that will work. And so what does normal MUC1 expression look like? Well, I told you that MUC1 is found in many epithelial tissues. Here's kidney, human kidney. And we've co-stained human kidney, a normal human kidney, from a normal individual, healthy individual, for EpCAM, which is just a marker of a cell surface, that's in red. And then green is staining with an antibody called HMFG1. That's a antibody that recognizes normal MUC1. And it shows us that normal MUC1 is found in green at the membrane of the distal tubules and the lumen of the kidney, lumen of the distal tubules, and then also in the lumen of the glomeruli. So it's found cell surface. That's important. And then if we look at where is TMF1 found? Oh, actually, I skipped a little bit here. We've moved to where TMF1 is found. I tell you that TMF1 in the kidney is not found at the cell surface. It's very punctate and intracellular. But if you look at where it colocalizes using a marker of a Golgi, it almost perfectly colocalizes in normal tissue. And this is important because this is where oligocostylation occurs in the Golgi. And so it's expected that we would find TMF1 expression in the Golgi of the normal human pancreas, as well as other normal tissues. But in cancer, TMF1 is found brightly expressed at the cell surface. And you can tell that here by how the green is outlining many of these tumor cells. That's in contrast to where we would find it in normal tissue. And this is actually a very common staining pattern. So we utilize the genetic sequence of the 5E5 antibody that recognizes that TMF1 peptide, that's glycoepitope, and we constructed a chimeric antigen receptor. And so that chimeric antigen receptor is presented here to the left, where we have the two variable domains of the antibody, and then we have a membrane anchor, a transmembrane domain. This one comes from CD8 alpha, and then we have an activation domain and costimulatory domain of that CAR. When we coat a plate, a tissue culture plate with peptides, much as we did for that antibody ELISA that I showed you recently, and then we throw onto those plates T cells that express these chimeric antigen receptors, either expressing the TMF1 specific CAR T cell or expressing the one that I told you about before, the CD19 expressing T cell that had gone into that patient who's now been in remission for 10 years. When we put those T cells expressing those two CARs onto these peptides, the TMF1 specific peptide releases cytokines like IL-2 and interferon gamma, those effector molecules that T cells produce when they're activated. They secrete those specifically to the TMF1. But if you look for the red, we don't see any red, right? That's because the red is the MUC1 without TM antigen. There's no secretion of cytokines, no recognition. So this antibody as a CAR has the same specificity that it did as an antibody. In addition, I'll point out that that white bar, that acylated sheep mucin, also has some cytokine secretion at high peptide concentrations. So it's at lower peptide concentrations, nothing, but at higher concentrations, there is some activity suggesting that this CAR can target TN antigen on other proteins. And will this work if you threw it into a patient, right? So would the CAR T cells be able to recognize the TN MUC1 positive tumor cells, but not recognize the MUC1 positive tumor cells, or the tumor cells that don't have TN antigen? Well, we tested that. We first, if you look to the right of the screen, we constructed a new cell line using a parental T-cell leukemia line called JERCAT. The parental number is E61. This line naturally expresses high amounts of TN-MUC1, but it's a T-cell leukemia, so it does not express CD19, which is found on B cells, not T cells. We made a new construct, a new lentiviral construct, where we use a truncated form of CD19, just the extracellular portion of CD19 and its transmembrane domain, a P2A ribosomal skip peptide, and then the gene for cosmic. Cosmic is mutated in JERCAT cells, and that is why JERCAT cells express high amounts of TN antigen, well, for TN-MUC1 and every other oglycosylated protein. And when we express that wild-type cosmic in these JERCAT cells, they now lose expression of TN-MUC1, and they gain expression of CD19 because of the truncated CD19. So when we culture both of these cells in the same well, we call them TN positive, that's the parental, and TN negative, that's the CD19 positive JERCAT cells. When we add to that well in increasing amounts, T cells that are not transduced, they don't kill either the TN positive, TN negative, they're not specific for JERCAT cells. When we add in T cells in increasing quantities that target CD19, we start to lose the CD19 positive, TN negative JERCAT cells, but those that are negative, TN positive and CD19 negative are fine. They're spared. And when we do the opposite, we add in T cells that target TN-MUC1, we lose the T cells that were TN-MUC1 positive, but not the cells that were TN negative and CD19 positive. So that tells us that these CAR T cells can differentiate from a hypoglycosylated cell, from a normally glycosylated cell in the same well, and presumably in the same tissue. And then we move forward to show that this can work in vivo. So we took 40 mice. These are NSG mice. They're non-SCID gamma. That means that they are immunodeficient mice. They do not have their own murine T cells or murine B cells. They also have some decreases in NK cells, some NK cell defects. They work really well to engraft tumor cells and engraft tissue like T cells. And so in these mice, we first, on day 0, injected 5 million JERCAT leukemia cells. These are the parental JERCAT cells. We've made them bioluminescent so that we can inject luciferin, like firefly luciferin, and view the tumor burden in these mice. And so this is an image 7 days after we had infused those JERCAT cells that shows us the tumor burden in those mice. And then 1 day after this, on day 8, we treated the mice with either PBS in group 1, non-transduced T cells, that just means regular T cells from a patient that have been expanded the same way as our CAR T cells, but they don't have a CAR, so they're not specific. That's a control group. And then we gave another group, group 3, CD19 specific CAR T cells. Now these are the parental JERCAT cells, so they're not CD19 positive. JERCAT cells is a T cell leukemia. And then in group 4 and group 5, we gave 2 different doses, 5 million or 10 million TN-MUC1 specific CAR T cells. And it's actually 5 million, 10 million total T cells. It's not CAR positive, about 50%-ish CAR positive cells in these groups. And then we did bioluminescence imaging of these mice serially, weekly, to verify the tumor burden. And what we see for the first 3 groups, the control groups, PBS, non-transduced, and CD19, is that the burden of the mice doesn't decrease. And actually we start to lose mice. They end up succumbing to paralysis from the tumor. The tumor invades their bone marrow, and then the mice end up with paralysis. And then in group 4 and group 5, what we saw was actually a reduction in tumor burden. These CAR T cells were working in vitro. They were killing the tumor, similar to what they did in the in vitro studies. And what's important here is that we know that mice and humans actually have very similar patterns of oglycosylation. Mice develop TN antigen, Mice have been shown to spontaneously develop tumors that express the TN antigen, and antibodies have been found that react against TN peptides in mice. And when you use CAR T cells in these mice, those CAR T cells can kill the tumor, but they don't kill the mouse. So that's important here for us as well, because we didn't see our mice die from treatment. And that means that this is potentially a safe target to put into humans, because they did not cause disease in mice, that they would likely not cause disease in humans. That gives us confirmation that these TN peptides may actually be more tumor specific than anything we've seen prior. And we can show that I showed you previously the antibody reacts to multiple different tumor types. Here we showed that the CAR T cells can actually recognize many different tumor types as well. So these are all cell lines, but there's cell lines from different tumor types like leukemia, pancreatic cancer, breast cancer. And then we measured the interferon gamma that's produced by CAR T cells, and the TMF1 CAR T cells here are in red. And you see interferon gamma produced by those CAR T cells, suggesting that they are reacting to the tumor. They're finding TMF1, they're getting excited, there's signal transduction, and then they start to make those effector molecules like perforin and granzyme B and IL-2 and interferon gamma. We moved forward with another tumor model. This time we did a pancreatic cancer model. Pancreatic cancer is a really aggressive cancer, and if we had a T cell therapeutic that could target pancreatic cancer, that could really impact the lives of patients. And so we injected one metastatic pancreatic cancer cell line, HS766T, into peritoneal mice. I'm showing you a representative example of one of those tumor inoculations. This is a mouse that had that tumor for about a month. And in dark purple, you can see the pancreas of the mouse. And then in the light purple, that's actually the tumor that was injected. You can see blood flow from the pancreas to the tumor. The tumor was actually attached to the pancreas in this mouse. And then below, we have immunohistochemistry will be stained for TMF1 with that 5E5 antibody. And you can see that most of the tumor had some speckles. It's not 100% ubiquitous expression of TMF1. So it's really interesting that when we treated these mice with our TMF1 CAR T cells, 3 weeks after the CAR, the tumor was gone. And mice survived 115 days after treatment, and no tumor came back. So that's really, really interesting. We think that it could be that we're looking at TNMUC1, but there may be other non-MUC1 positive cells that express TN antigen that the CAR T cells are recognizing at lower levels, right? TN, other peptides, and they're clearing that tumor. But that may be possible. However, that's just a speculation. We also were really interested to know the potential for targeting of other types of human cells. And so some human cells we were able to acquire, primary human cells, we were able to acquire and sorry, I'm just checking my time. We were able to acquire the primary human primary human cells that some that aren't expected to express MUC1, like cardiomyocytes and aortic smooth myocytes, osteoblasts, but others that are expected to express MUC1, like renal epithelial cells and renal cortical epithelial cells. When we treat them with our CAR T cells, either targeting CD19, now an FDA-approved CAR T cell product, or for our TMF1 CAR T cells, we don't see any increased cytotoxicity against those primary cells than we saw with CD19 CAR T cells, suggesting that our CAR T cells may be as safe against these particular tissues as CD19 CAR T cells. And so that's really encouraging. And with that, we actually started a clinical trial. And so this was opened on July 18th, 2019. And it is a trial at UPenn, as well as several other institutions. It's a multi-center trial now for 6 indications. It's sponsored by a company called Community Therapeutics. And there are 6 different indications: non-small cell lung cancer, ovarian cancer, fallopian tube cancer, triple negative breast cancer, multiple myeloma, pancreatic ductal adenocarcinoma. And if you are interested, you can you can put your phone over that Scan Me QR code, and you can get a link on your phone to the website to find out more information about this trial. And so with that, I think I'm gonna show you a couple more updates where we're trying to improve the activity of our CAR. So we use a stress test, which we put the CAR T cells into a lot of tumor until the point where we find a dose of CAR T cells that no longer can clear 100% of the tumor. That's what you see here in the second video that I just showed you. The tumor is labeled with a red nuclei. So our non-transduced T cells have no efficacy. And dead tumor turns green in these assays. So in this video, you see that our red tumor, when added with these T cells, starts to develop green. That's a signal of death, where the tumor is dying. You see at the end of all of these videos, either ineffective, killing, worse than our parental, in which we express, in addition to our CAR, we express a cytokine, in this case, IL-7, IL-12, IL-18, or 3 different cytokines that we're not disclosing yet. And what we see is that some cytokines, like IL-7 and IL-18, and the 3 undisclosed, do a really good job at enhancing the activity of the tumor, activity of the T cells to clear the tumor, but others, like IL-12, do not. And so, so we're really interested in following up on this and combining these cytokines with our CAR T cells, either exogenously, so adding them to the patient in order to improve activity, or to incorporate this in our CAR design, and so we get a great benefit from the CAR T cells. And I'm running out of time. The last thing that I want to share with you is that other work we're doing stretches the limits of T cell biology. And so this is work by Tiffany King in my lab, a postdoctoral fellow. She has developed CAR T cells that, unlike normal CAR T cells, which have all traditionally targeted a molecule expressed on a T cell surface. I told you CARs can only recognize cell surface antigens. That's actually not true anymore. So Tiffany has engineered CAR T cells to recognize extracellular matrix proteins. So these are proteins that are not even associated with the cell surface. They're secreted by stromal cells, secreted by tumor cells, and they're present in the extracellular matrix outside of the cell. Well, when CAR T cells target these extracellular matrix proteins, in this case EDB, I'm going to skip, they kill tumor. And so this is an example of fibronectin-specific CAR T cells targeting a prostate cancer. And then the red line is our TMF1 CAR, and then the blue line and green lines are another TN targeting CAR, but this is TN fibronectin. It's also known as 3CS fibronectin, and these CAR T cells are just as lytic as our TMF1 CAR T cells, even though they aren't targeting the cell surface. We're investigating ways in which this activity occurs. How does a T cell kill when they don't target the cell surface? And so we're looking at mechanisms of killing. And this is more data to that effect. Here's some animal data that shows you that these CAR T cells work really well to reduce tumor burden in mice that are carrying this subcutaneous prostate cancer, and it works really fast. So this is a very interesting target for us. And the T cells infiltrate the tumor just as well as those that target the tumor surface. And interestingly, they have some stromal alterations in the tumor that may be specific to this type of T cell that targets a fibronectin. While another type of fibronectin CAR that we've also worked on for several years doesn't work as well as these TN fibronectin CAR T cells. So we're really interested in the combination of a T cell that can target an extracellular matrix protein and a T cell that can target a cell surface protein, and can that overcome more tumor escape mechanisms. So with that, I'll summarize that we've shown you that T cells targeting alternative abnormal glycoforms of tumor-associated proteins enhance the specificity of cancer targeting. We've shown that ectopic expression, that's in these videos that I showed you, of unique effector molecules like cytokines can enhance CAR T cell activity. However, the temporal nature of ectopic expression must be critically evaluated. That's why IL-12 doesn't really work as well. And then lastly, T cells targeting extracellular matrix proteins of a tumor microenvironment are lytic. They can kill, and particular therapies can enhance T cell infiltration and reduce tumor control. This doesn't This isn't true for every T cell targeting extracellular matrix protein like that other EDB fibronectin. We think that the 3CS or TN fibronectin is superior. With that, I'd like to thank my lab, former members of my lab who's done this work, collaborators in Copenhagen and Chicago, collaborators here at the University of Pennsylvania, my funding, and if you're interested to follow us on Twitter, as well as more information about our clinical trial. And with that, I thank you for your attention.
Right. That was a fabulous talk by Dr. Posey. And the activism that he was talking about as well was amazing. I hope that everyone enjoyed it as much as I did. Well, now let's get to the next talk. We have the pleasure of Dr. Donita Brady speaking today. She received her PhD in pharmacology from the University of North Carolina at Chapel Hill. Go Tar Heels, that's my home state. Notably, she then went on to do a postdoc in Dr. Christopher Counter's lab at Duke. Wow, in my home state again. And she studied how copper's role is important for cancer progression, and is now a Harrison McCrae Dickinson MD and Clifford C. Baker MD Presidential Assistant Professor of Cancer Biology to further study copper metabolism in cancer and tumorigenesis. Please welcome Dr. Donita Brady.
Hi, my name is Donita Brady. I'm the Presidential Assistant Professor of Cancer Biology at the University of Pennsylvania and the Perelman School of Medicine. And I'd first like to thank the organizers of the Black Queer Town Hall in STEM for giving me the opportunity to share with you some of our research on the interplay between copper homeostasis and kinase signal transduction that we try to leverage as a new cancer therapy. Normal physiology relies on the orchestration of complex processes within cells, between cells, and among cells in their environment to maintain proper tissue homeostasis. And to enable the precise coordination of these cellular processes with dynamic spatial and temporal control, evolutionarily conserved signal transduction pathways serves as a means to both integrate and distribute information. And proteins within these signaling pathways function as nodes to both sense and respond to inputs, and in turn, facilitate or regulate outputs. Unsurprisingly, aberrant activation of signaling initiators or inactivation of signaling repressors generates an imbalance in normal cells and disrupts the tight control signal transduction pathways typically have on cellular processes that, when dysregulated, drive the hallmarks of cancer. And decades of research has revealed the complexity of oncogenic cellular signaling networks and their exceptional vulnerability as targets in cancer. My lab studies protein phosphorylation, and it's the most common form of reversible post-translational modification, with an estimated 50% of all proteins undergoing phosphorylation. The phosphorylation state of any given protein is controlled by the coordinated action of specific kinases and phosphatases that add and remove phosphate, respectively. While there are at least 518 kinases and 156 phosphatases in the human genome, recent pan-cancer analysis of the major cancer types revealed that 91 protein kinases could be considered driver oncogenes. And this occurs through either point mutations, gene amplification, or chromosomal translocations. Of these 91 driver protein kinase oncogenes, many of them have FDA-approved small molecules, and only 4 are orphaned in the sense that they have no known small molecule intervention. And while small molecule therapies against these aberrantly activated protein kinases has revolutionized cancer therapy, there are still several challenges. First, the emergence of resistance. Second, tumor cellular heterogeneity. And third, these undruggable targets. And this really limits their widespread use as therapeutics in cancer. One of the most well-illustrated forms of acquired resistance is in the context of BRAF mutation-positive melanomas that typically respond to inhibition with MAP kinase pathway therapies. As many of you know, in 2002, a group searching for mutations in the canonical members of the RAS, RAF, MEK, ERK signaling cascade in a large panel of common cancers revealed that 60% of melanomas carry an activating mutation in the gene encoding the serine-threonine kinase BRAF. And a follow-up to this breakthrough study identified that BRAF mutations are present in precancerous nevi, indicating that this mutation is indeed a driver of the initiation of melanoma. And so because of this, many pharmaceutical companies went after small molecule inhibitors of oncogenic BRAF or its downstream kinases MEK1 and 2. As I'm showing you here with this at the bottom of the screen, there is very good response to BRAF pathway inhibition. Specifically, this patient presented with melanoma deposits on the chest and was given the BRAF inhibitor vemurafenib for about 15 weeks. And within those first 15 weeks, you could see almost complete resolution of those lesions. However, about 8 weeks later, the patient represented with relapsed disease, where you could see that the same lesions had come back. And this was due to reactivation of that canonical MAP kinase pathway, specifically activation of MEK1 and 2. And so this really illustrates the need to understand novel modes of kinase regulation in order to forestall this resistance that can occur. And since the activity of kinases is governed by a multitude of inputs, such as nutrients, each of these inputs becomes a potential therapeutic vulnerability. And my lab is interested in the interplay between oncogenic kinase signal transduction and nutrient homeostasis in order to leverage those interactions for the treatment of cancer. Unlike organic metabolites that can be synthesized and degraded via enzymatic reactions, metals shown here at the top in terms of the periodic table are unique micronutrients that can neither be created nor destroyed. Therefore, proper dietary intake of these metals and strict homeostatic mechanisms to monitor their abundance are vital. Our lab and others are interested in this emerging paradigm where transition metals actually function as signaling molecules themselves by directly interacting with biomolecules that then govern and coordinate biological activities in response to stimuli. For example, we established that the MAP2 kinases, MEK1 and 2, which are downstream of oncogenic BRAF, are copper-binding proteins, and that copper can directly impact MAP kinase pathway activation. Specifically, as shown here on the right, as you increase the amount of copper sulfate, and in a MEK1 kinase assay, you see increased phosphorylation of the target ERK2. But if you spike in the copper chelator tetrathione, which is an investigational drug for the copper overload disease, Wilson disease, you see inhibition of the pathway. And so this really suggests that there's other ways to regulate this critical signal transduction pathway. Given this newly discovered signaling paradigm and the dependence of BRAF mutation positive cancers on MEK1 and 2, we previously investigated whether decreasing the influx of copper via the copper transporter CTR1 could impact BRAF-driven tumorigenesis. We found that overexpression of oncogenic BRAF V600D actually required copper transport through CTR1 for robust tumorigenesis, as shown on the top right. And in follow-up work, we demonstrated in the BRAF-driven genetically engineered mouse model of metastatic melanoma that the copper chelator TTM could dampen tumor growth kinetics alone and in combination with the clinical MAP kinase inhibitor vemurafenib by specifically reducing MAP kinase signaling. Further, TTM inhibited the growth of MAP kinase inhibitor resistant forms of melanoma, as shown on the right. Thus, copper chelation using TTM may represent a component of a rationally designed strategy to limit MEK1/2 activity in BRAF-driven tumorigenesis. But when I started my lab at Penn, we wanted to think a little bit bigger. And so we've been thinking a lot about copper-mediated kinase signal transduction and whether or not this might be a generalizable vulnerability in cancer. And one of the major questions that we had in the beginning is whether or not there were other protein kinases that could be regulated by copper and what other cellular pathways or processes may be impacted by reductions in copper availability. The real driving force behind these long-term research goals is the hypothesis that kinase activity is in part dependent on metals in order to ensure that cellular processes essential for proliferation are restricted to the optimal growth conditions. Specifically, the amount of copper that's available will coordinate whether or not growth can occur. And so to date, we have utilized multi-tiered, unbiased screening approaches to identify 66 other copper-dependent kinases, each representing an unknown contribution of copper to a signaling event or a cellular process. One of those approaches we utilized to define the landscape of copper-dependent kinases was to leverage our previous elucidation of the amino acids that were important for binding to the copper within MEK1 and 2 to identify 10 kinases with significant in silico sequence similarity to MEK1 and 2. Of interest to graduate student Tiffany Zhang and postdoctoral fellow Jessica Pacima in my lab were the autophagy signaling kinases ALK1 and 2. And as shown at the top of the screen, they both share identical amino acids previously identified to be important for MEK1/2 copper binding. Therefore, we tested and found that both ULK1, shown here, and ULK1 and ULK2 bind to a copper-charged resin, but not a metal-free resin control or iron or zinc-charged resins. In addition to that, we wanted to investigate the impact of copper binding to ULK1 and 2 function. So we tested whether copper is directly required for ULK1 and ULK2 kinase activity. In a series of in vitro kinase assays, we found that increasing the concentration of copper chloride could increase the amount of ULK1 phosphorylation of its target HG13, while increasing the concentration of the copper chelator TTM could inhibit the ability of ULK1 to target HG13. And so these results really show us that ULK1 and 2 bind to and require copper for their kinase activity. So what are ULK1 and 2? These are well-established downstream targets of the protein kinase mammalian target of rapamycin, or mTOR. When associated with the rapamycin-sensitive mTOR complex 1 or mTORC1, mTOR controls cellular energetics by driving anabolic processes such as lipid, nucleotide, and protein synthesis, and suppressing catabolic processes like autophagy. mTOR kinase activity is blocked in response to amino acid deprivation or low cellular ATP levels, so specifically when nutrients are withheld. This in turn relieves the inhibitory phosphorylation on ULK1 and 2 and initiates the formation of an autophagosome. And this autophagosome continues to grow, and it degrades and recycles proteins, lipids, and organelles to restore nutrient balance in the form of building blocks for metabolic processes. Specifically, active ULK1 translocates to the ER membrane with its constitutive binding partners ATG13 and 5200 to drive PHAGA4 formation by phosphorylating core components of the class 3 PI3 VPS34 complex. This ULK1-mediated phosphorylation of members of the VPS34 complex stimulates its kinase activity and converts phosphoinositol to PI3P. This generated PI3P serves as a docking site for 5-domain-containing proteins like DFCP1 or WIPI1 that recruit the ubiquitin-like conjugation complex ATG5, 12, and 16 to lipidate LC3B. And so this is why we often use lipidated LC3B as a readout for autophagy induction. And so we next wanted to understand whether or not copper was acquired for autophagy induction. And so to investigate this relationship between copper and ULK1 activity in mammalian cells, we created copper replete and copper deficient cells by overexpressing the high affinity copper transporter CTR1 or an empty vector into a mortalized CTR1 knockout mouse embryonic fibroblast. And we determined whether or not copper levels were restored by utilizing the CF4 copper probe by Kiss Train's lab at University of California at Berkeley. What we found is re-expression of the wild type transporter could restore copper, labile copper levels, while the CTR1 knockout cells have about a 50% reduction in copper availability. We then next wanted to ask, what happens if we expose those CTR1 wild type or knockout cells to low energy states, or withdraw amino acids? The data that I'm showing you here is in the context of amino acid deprivation, which will inhibit TOR and activate ULK1 to induce autophagy. We also add baphlomycin, which prevents the degradation of the autophagosome and the lysosome so that we can see the amount of LC3B2 that's generated. Of importance, when we clamp cells with baphlomycin, we see an increase in LC32 levels in our wild type cells, but we see reduced amounts of LC32 in the CTR1 knockouts. Similarly, when we withdraw amino acids, we see an increase in LC32 levels, but not in the absence of the copper transporter CTR1. And so this really shows that the loss of CTR1 significantly reduced LC31 processing and thus autophagy induction. And since our findings suggest that copper is really required for ULK1/2 dependent signaling and autophagy flux in response to mTOR inhibition, we hypothesize that copper levels may actually, in fact, fluctuate in an acute manner after autophagy induction. And so we utilize the same CF4 copper probes in these mouse embryonic fibroblasts to look at what happens to copper levels when we withdraw amino acids. In our control cells, when we're imaging every 5 minutes for about an hour, we saw no real changes in the labile copper probe as shown by CF4. However, when we withdraw amino acids and look every 5 minutes for about an hour, we see that there's this increase in intracellular copper availability. This is either coming from extracellular or intracellular stores of copper. When we evaluate this statistically, we find that there's a significant increase in the labile intracellular copper pool when we withdraw amino acids. And thus this requirement for copper for efficient autophagy flux is driven by fluctuations that support ULK1/2 kinase activity. In agreement with the fact that amino acid withdrawal prevents mTOR inhibition of ULK1 and 2 and increases kinase activity, we found that if you withdraw, if you deplete CTR1, you're no longer able to sustain this increase in ULK1 kinase activity in the context of amino acid deprivation. And so this really shows that copper is necessary for ULK1 activity downstream of amino acid deprivation. As cancer biologists, we were really interested in translating this idea to the context of an in vivo scenario that required autophagy. Increased levels of basal autophagy in some cancers represents a vulnerability to autophagy inhibition by blocking lysosome function. And importantly, oncogenic KRAS G12D, which is the most common oncogene in lung adenocarcinomas, also depends on efficient autophagy for tumor maintenance. And this has really been shown by Eileen White's group. And they showed that deletion of the critical autophagy component HG7, which is actually really important for the lipidation of LC3B, converts KRAS G12D driven adenomas and adenocarcinomas to oncocytomas, a benign tumor type characterized by an accumulation of dysfunctional mitochondria. What this also showed is that there was an extension in the time that the survival of animals and the absence of autophagy in this KRAS-driven model. And so we were really interested in whether or not CTR1 deficiency could impact autophagy induction. This is really critical in thinking about this in the context of KRAS mutations. KRAS is known to activate the canonical MAP kinase pathway. And so people have often tried to either target KRAS or MAP kinase pathway components as therapeutics in this disease. And it was recently published by two separate groups, specifically Chanda and Martin McMahon's group, to show that inhibition of the canonical MAP kinase pathway or KRAS itself leads to protective autophagy in cancers. And so you need dual inhibition of both the canonical MAP kinase pathway and autophagy in order to block KRAS-driven tumor growth. And so what we had shown is that CTR1 is both important, or copper transport specifically, is both important for MAP kinase pathway activation and ULK1/2 driven autophagy. And so we reasoned that CTR1 loss would block both of these processes. So to do this, we collaborated with members of David Felser's lab to use the same lox-out-blox KRAS G12D and P53 FOX model used by the White group. We also had lox-out-blox luciferase driven by the ROSA26 locus. And this allowed us to monitor tumor growth kinetics over time. Specifically, these mice were given intratracheal administration of lentivirus expressing pre-recombinase, Cas9, and guide RNAs targeting either beta-galactosidase or CTR1, and tumor growth was monitored over time by bioluminescence. What we find is that CTR1 loss leads to a significant reduction in tumor growth kinetics over time. And if you look at this at a fixed time point, specifically 16 weeks, you see that there's about a 50% reduction in tumor burden. And so this really shows the CTR1 loss is correlated with reduced in vivo tumor cell proliferation. Not surprisingly, when we looked at these lesions at a fixed time point, we saw that genetic ablation of CTR1 decreased MAP kinase signaling as read out by the phosphorylation of ERK1/2. And this is definitely critically important for blocking lung tumor development. Simultaneously, the loss of CTR1 also reduced LC3 positive staining in these lung tumors driven by oncogenic KRAS, suggesting that autophagy was blocked. But to look at this more directly, we also quantified the number of autophagosomes that were formed by electron microscopy. And here we saw a significant reduction in autophagosome number and an increase in mitochondrial number. And thus the loss of CTR1 mitigates KRAS G12D mediated autophagy that is necessary for lung tumor growth. To connect this molecularly, we next wanted to interrogate whether or not the contribution of copper binding to ULK1 was required for kinase activity. So we made point mutations at the same residues within MEK1 and 2, which were able to reduce its ability to bind to copper, and were able to create a copper binding mutant of ULK1. And this mutant of ULK1 has reduced ability to phosphorylate ATG13, suggesting that binding of copper to ULK1 is required for its kinase activity. To next investigate this in the context of endogenous oncogenic KRAS tumorigenesis, we knocked out the endogenous ULK1 and 2 via CRISPR-Cas9 and mouse KRAS mutant lung adenocarcinoma cell lines. As expected, the knockdown of ULK1 and 2 in these cells led to reduced LC3-2 levels and led to reduced phosphorylation of ATG13. We were able to rescue this by re-expressing ULK1 wild type, but not the copper binding mutant. And so this really shows that binding of copper to ULK1 is required for autophagy induction and signaling in these KRAS G12D driven lung tumors. We then took those same cell lines and injected them into immunocompromised mice to look at tumor growth over time. We found that knockout of ULK1 and 2 led to a similar reduction in tumor growth over time as compared to ATG5, which is also a component of the ubiquitin-like complex that's important for lipidating LC3B. So ULK1 and 2 are required for this KRAS-driven tumorigenesis. We could rescue this by re-expressing ULK1 wild type, but not a copper binding mutant, suggesting that oncogenic KRAS-driven tumorigenesis does require ULK1's ability to bind to copper. So in summary, we discovered that copper binds to and is required for ULK1 activity, thereby serving as a critical input for ULK1/2 dependent autophagy. Dual targeting of MAP kinase signaling and ULK1/2 driven autophagy by limiting copper availability and therefore kinase copper binding decrease oncogenic KRAS driven tumor growth and survival. These findings chart new ground in nutrient signaling, cellular energy homeostasis, and metabolic vulnerabilities in cancer, and further establish copper as a signaling molecule, define the molecular basis for a new copper-dependent cellular process, and exploit copper-dependent kinases to target oncogene-driven dependencies. A lot of this work was done by Tiffany Zhang, a graduate student in my lab, and Jessica Basimo, a postdoctoral fellow in my lab. We collaborated with members of David Felder's lab, specifically Andrea Gudial and Michelle Ticini, to do this work. In conclusion, I'd like to thank the people who have supported us throughout our time doing this research, our many collaborators here at Penn, at my formal institution, Duke, and across the country and world. And I'd like to also thank our funding sources, and I'd be happy to take any questions at the end of the session.
Dr. Brady, that was an amazing talk. Our next speaker is Dr. Michael Boyce. He received his PhD in cell biology from Harvard Medical School. He then went on to a postdoctoral fellowship in Dr. Carolyn Bertozzi's lab, elucidating glycol metabolitic pathways, and is now an associate professor of biochemistry at Duke University studying glycobiology. Dr. Boyce has also dedicated a significant amount of work to making STEM more equitable for all groups by co-directing the Maximizing Opportunities for Scientific and Academic Independent Careers program, or Mosaic, that aims to facilitate the transition of scientists from diverse backgrounds to tenure track research faculty positions. Please welcome Dr. Michael Boyce.
My name is Mike Boyce. I'm an associate professor in the biochemistry department at Duke University. And it's my honor to speak with you today about both my science and the allyship work that I've been doing as a faculty member at Duke. So I want to start by thanking the organizers for this opportunity to speak with you today. I'm really sincerely honored and humbled to be included as an ally in this fabulous Black, Queer, and STEM event. I think it's great that it has come together the way it did, and I'm so glad to be a part of it. Per the recommendations or instructions from the organizers, I'm going to start with a research overview of the work in my lab, which is the title at the top, the cell signaling through protein glycosylation, and then offer just a few thoughts of my own, my personal opinions on the role of allyship in addressing equity and inclusion as a practicing biologist in academia. So my lab studies glycobiology, which is sort of a subset of biochemistry or cell biology, maybe unfamiliar to some cell biologists or biochemists themselves. So I just wanted to start with a quick overview of what glycobiology is in general. Glycobiology, I would say, it could be defined in many ways, but an easy operational definition is to say that it's the study of how living cells and organisms make stuff out of sugars. So carbohydrates are chains of sugars, monosaccharides that are strung together in a number of enormous number of chemically complex ways. And this is a feature of all life on Earth. All organisms and all cells on Earth have glycans, or chains of sugars that modify their proteins and their lipids to control various aspects of the cell. So this diagram here shows a cartoon of a diverse range of glycoconjugates, or sugar chains connected to other molecules like proteins and lipids that are made in a typical animal cell, like a human cell. And these really influence all aspects of cell physiology in mammals. So it's tremendously important from a basic cell biology perspective, but it's in some ways more challenging to study than things like DNA or RNA biosynthesis, protein synthesis, because those processes are templated. DNA makes RNA, RNA makes protein, whereas glycans, these sugar chains, are not synthesized from a pre-existing template, and therefore it can be more challenging to study with sort of traditional genetic or biochemical methods. So we're particularly interested in the role of cell signaling through protein glycosylation. So how cells use these carbohydrate structures to transmit information either internally or between cells. I'll also say that, believe it or not, this sort of Lucky Charms looking panel of cartoons on the right with various monosaccharides like glucose and galactose, this is a relatively standard nomenclature in the field of glycobiology, and I'll be using it throughout the talk. So a few slides have chemical structures that depict glycans, including the next one coming up, but most I'll be using these shapes to depict the complex glycans. So unlike DNA or RNA or proteins, which have a sort of bead on a string structure where nucleotides or ribonucleotides or amino acids are strung together in a linear polymer, glycans can exhibit enormous chemical complexity because they can include branching and they can include different stereochemical connections. So this might be a little bit of inside baseball for people who are more chemically inclined in the audience, but I just wanted to show two structures of typical M-glycans, meaning a glycan connected to an asparagine side chain on a protein, or an O-glycan connected to hydroxyl side chain, an oxygen atom, hence the term O-glycan. from either a serine or a threonine side chain on an amino acid. And I think you can appreciate just from this, just the shock and awe aspect of how complicated these structures are, that there's really a mind-boggling array of chemical structures that glycans can adopt through not only a number of different building blocks, but also different ways that they can be connected to each other. And these structures matter because proteins or other biomolecules can tell the difference between subtle variations in these structures. So it really is a fascinating sort of chemical space and biological problem to study the function of these molecules. Now, you might remember from high school biology or college biology that proteins that get glycosylated and then secreted outside the cell usually begin their life in the endoplasmic reticulum and then the Golgi. This is a cartoon from the Essentials of Glycobiology textbook, which is available online through the National Center for Biotechnology Information library site, if you want to look at it. And it depicts different classes of glycans, like going left to right, the N-glycans and O-glycans I just mentioned, some other slightly more exotic versions. And it just sort of shows in assembly line fashion how proteins that are destined to be modified with these glycans move through first the endoplasmic reticulum and then the Golgi, and can get secreted to the cell surface, the plasma membrane, or entirely outside the cell, for example, into the bloodstream. And as they move through this process, they acquire more and elaborated glycans. So this is just a cell biological overview, kind of shows you the really interesting and elaborate process that these glycans go through during their biosynthesis. Another interesting glycan that is different from this is what's referred to as O-glycnac. This is the addition of a single O-linked N-acetylglucosamine, or glycnac, which is the sugar depicted in blue here. This is actually an intracellular form of glycosylation, meaning it's inside the nucleus, mitochondria, cytoplasm. So it's topologically distinct from the other glycans I just mentioned. It's not outside the cell, instead it's inside the cell. And I just wanted to sort of shout out to it here because several projects in my lab focus on this form of glycosylation, although it won't be the subject of today's talk. So I sort of made this point earlier, but glycobiology really influences all aspects of cell physiology, from transcription inside the nucleus to cell-cell adhesion, host pathogen interactions with bacteria or viruses interacting with sugars on the surface of the cell. So for a basic cell biologist like myself, it's a really rich problem and field to work in and study because it is fundamental to essentially all life processes. In mammals, but really all organisms. In addition to that, glycobiology is integral to the study of nearly all human diseases. This is a diagram I like to show to illustrate this point. It might have a little more detail than is necessary, but what you're looking at here is inside the endoplasmic reticulum, the various enzymes at each step that are used to build up the N-glycan made up of those green mannose and blue circle glucose and blue square glycnac sugars. And this is a highly regulated process that goes stepwise, where one sugar is added, then another, with a particular branching pattern. The point I want to make with this slide is that if you look at all of the red X's on this diagram, everywhere there's a red X, there's a known human disease where that enzyme is mutated. These are referred to as congenital disorders of glycosylation, or CDG, which is the abbreviation you can see there next to the X's. And this just really shows you how the intricate process that goes into glycosylation is so important for human health and human disease. Similarly, more familiar diseases like metabolic disorder, diabetes, cancer, neurodegeneration are also all characterized by disturbances in normal glycosylation inside cells. So therefore, it's part of disease processes that are probably familiar to all of us. And some therapeutics that are very important, like monoclonal antibodies that are used in the clinic, are themselves glycosylated. So those proteins are glycoproteins, and the sugars on those proteins are important for their function as drugs. If you're more into evolution than in biomedicine, there's good news for you in glycobiology as well. As I mentioned earlier, glycosylation it's really ubiquitous in all organisms on Earth. This is just a chart that illustrates that. So the diamonds you're looking at, the black diamonds are glycoside hydrolases. These are enzymes that remove glycans from proteins. The magenta squares are glycosyltransferases. These are enzymes that add glycans to proteins. And if you look in eukaryotes at the top, or bacteria in the middle, or archaea, these are the three main groups of life on our planet. What you can see is that as genomes increase in complexity with the number of genes illustrated on the x-axis in each of those boxes, what you see is an increase, corresponding increase in the complexity of the glycosylation machinery, suggesting that the glycosylation and its regulation is really a feature of life that is universally conserved across the planet. I mentioned earlier that glycobiology is important to biomedicine. I'm grateful to say that the National Institutes of Health, or the NIH, which is the agency that funds most biomedical research in the US, has recognized this. If you're a biologist listening to this and you want to learn a little bit more, I would direct you to this glycoscience website that the NIH Common Fund has. Lots of good resources there for both the public, as you can see on the left, and also scientists who are interested in this field. Although I work on sort of cell biology and human biology, disease, and health, glycobiology, I just want to point out, is really integral to other fields as well. Two examples. One is sustainable energy research and development. So on the left, you're looking at a cartoon from this report called Transforming Glycosciences, issued by the National Research Council. This is a cartoon of the cell wall of plants. So as you probably know, there's great interest in the renewable energy field and being able to ferment the cellulose and the pectin in the cell wall in order to have renewable sources of fuel to replace fossil fuels and to improve the outlook for the climate on the planet. The cell wall of plants and similar organisms is all carbohydrates. And so being able to understand how to release the carbon stored in cell walls and plant tissue in a way that is scalable and efficient to produce fuels and other fine chemicals is a major area of interest where glycobiology plays a role. And then finally, glycobiology is also integral to research and development in materials sciences. Here's two more examples from the NRC report. One is a flexible LED screen, which is created from a glycopolymer, a polymer of sugars put together. And at the bottom are stents created from cellulose, a bacterial cellulose in this case, not from plants, that are used in vascular surgery to provide relief to patients with blocked blood vessels. So distinct even from understanding how living organisms work, there's opportunities to harness the material properties of chains of sugars, glycans, and glycoconjugates for material science. So coming back to the overview slide, and to go to transition into the section of the talk about one project from my own research lab. Here, what you can appreciate, I think, even just looking at this cartoon, is that there's a lot of different sugars that can be used to make up glycoconjugates in mammalian cells, and they can be connected in different ways, as I mentioned earlier. Now, these sugars aren't just used to make these glycans as free sugars, the way they're depicted in the rectangle legend on the right side of the slide. Instead, they are used in a form of molecule called nucleotide sugars. So this is essentially a sugar like the yellow square or the blue square connected to what's called a nucleotide, which is a chemical moiety that's also common to DNA and RNA. So this nucleotide sugar precursor, you could say, is what enzymes use to attach sugars onto other molecules like sugars, proteins, lipids. This slide depicts the biochemical pathways through which cells can biosynthesize and interconvert different nucleotide sugar species. So in other words, this is the way the cell creates the full range of nucleotide sugars that it needs in order to build those glycans. The details on this slide are not super important. The point I want to make with it actually is that this is intricate and complicated. So the different nucleotide sugars can be made from different sources, including the nutritional sources, sugars that you eat, and then they can be interconverted into each other, as you can see from some of these bidirectional arrows. Now, our lab was interested in these pathways in general because although we understand them at the level of these arrows shown here, the sort of biochemical connectivity between these pathways, almost nothing is known about how cells regulate the flow of material through these pathways. So if a particular nucleotide sugar is high or low in the cell, or more is needed to biosynthesize a particular form of glycoprotein, how do cells control that sort of thing? Or under stress conditions or nutrient conditions where a certain sugar, let's say galactose, is very high in a meal that you or I have just eaten, how do cells process that and know how to direct the flux through these pathways so that they can remain in balance with the nucleotide sugar pools they need to perform all their functions? Surprisingly, very little was known about that when we began this project. So the work I'm going to show you about today was pioneered by Dr. Alex Broussard, who's on the right here. He graduated from the lab last year and is being continued now by Alicia Florwick, who is a graduate student in my lab. So Alex initially started to attack this problem by picking a model enzyme that's involved in nucleotide sugar biosynthesis, and then wanted to study it and ask what happens in the cell if we disable it in order to understand its normal function. So the one he chose is referred to as galactose-4 epimerase, or GALE for short. And GALE is a good model enzyme to choose for this problem because it balances the levels of 4 critical nucleotide sugar pools in the cell. So for aficionados, I'm showing the chemical structures here. UDP is the abbreviation for the nucleotide sugar, or the nucleotide portion of the nucleotide sugar molecule. And then the sugar is depicted in its chemical formula. So what you can see in the top left is UDP N-acetyl galactosamine, or GalNAc. And GALE can convert that back and forth to UDP glycnac, or N-acetyl glucosamine. Similarly, on the bottom, what you can see on the left is UDP galactose. Galactose, as you might know, is one of the 2 sugars that makes up lactose, like in dairy products. And it interconverts that with UDP glucose, glucose being the other sugar in lactose. Now, if you are not interested in the full chemical structures of these, we can also abbreviate these same nucleotide sugars with the cartoons that I showed earlier. GalNAc is the yellow square, glycnac is the blue square, galactose is the yellow circle, glucose is the blue circle. To go back to this slide for just a moment, I'm going to point out where GALE is on this chart. These red circles here show the 2 interconversions that I just showed you on the previous slide. And I think it's useful to look at it here as well because it sort of illustrates the point that GALE is a central node in nucleotide sugar metabolism. If you were to disable these steps here, you could follow the pathways along and notice that the chain is broken and interconverting different nucleotide sugars to each other. And so we believe it's an interesting model enzyme to study for that reason. So one of the first things Alex did was, in order to ask what the normal function of GALE is in cells and whether its balancing of nucleotide sugar pools is important for physiology, an early thing he did was to use CRISPR, a genome engineering tool that was the subject of the Nobel Prize just this past month, actually, to Emmanuel Charpentier and Jennifer Doudna, to delete the GALE gene in human cells. So he did this in multiple human cell lines. I'm just depicting one here. And what you're looking at in this slide are data from what's called a Western blot to detect proteins. The black blobs in the control cells in the top rectangle there are the GALE protein. And then if he uses different CRISPR, what are called guide RNAs, single guide RNAs or sgRNAs, we have 3 different ones to target the GALE gene. You can see that Alex successfully deleted GALE in all 3 of those cases. Each sort of lane, each vertical lane in this Western blot depicts a different clone that he derived from these genetically manipulated cells. Tubulin in the bottom box there is a protein that is present in all the cells, and that shows we haven't completely screwed up the cells. We have even loading of tubulin in all those cases. Okay, so Alex successfully deleted GALE in these cells. What might be the consequence? First, at the level of the nucleotide sugars, we saw some profound defects when GALE was absent. So first, what Alex did was to grow clones, either control clones, which he called A or B, or GALE-deleted clones, 1A with 3B here on the bottom in the x-axis. First in mannitol. So mannitol is just an osmolate control. It's a non-metabolizable sugar that he can add that will become important in just a moment. So this is sort of control normal conditions where you culture cells in petri dishes. So if you look at the blue data on the graph, you can see that the levels of, in this case, UDP galactose, the yellow circle, the levels of that particular nucleotide sugar are a little bit depressed in cells that are lacking GALE compared to the control clones. And that's to be expected because it's not able to be interconverted from UDP glucose properly. However, then if you feed the cells galactose, which are the red data here, now what you'll see is that the GALE-deleted cells hyperaccumulate UDP galactose. They're unable to metabolize that galactose kind of from the cell culture version of a meal when you feed them external galactose and then convert it through those nucleotide sugar pathways to make other nucleotide sugars and balance things out. The control cells do that fine, and you don't see a huge rise in the red UDP galactose in the control cells in the left two examples, but on the right, you see this hyperaccumulation of this species, which could be toxic to the cells. And I'll also note that the y-axis there has an axis break just for convenience on this PowerPoint slide. So you can see that you're really looking at, you know, a 10-fold increase or more in this particular metabolite when GALE is missing. So as you might imagine, this could have consequences. These imbalanced pools of building blocks could have consequences for the glycoproteins that the cell is able to make. And indeed, that proved to be the case. So some examples of data that Alex and Alicia obtained to show that are as follows. First, he used a technique called flow cytometry, which basically just uses proteins to give you a fluorescent signal of what kinds of sugar chains are on the surface of cells that you're looking at. The particular disaccharide or two sugars, moiety of two sugars that we're looking at in this data is depicted on the left. This is a sialic acid that's an alpha-2,6 linked to a galactose. There's the structure again for people who are interested, but in the cartoon format, this is what it looks like. It's a purple diamond and a yellow circle. So using a protein that can detect this diamond circle pattern on cell surfaces, Alex analyzed both control and GALE cells. So what you see here is a histogram where he's counting cells on the y-axis, and then on the x-axis, the fluorescent signal from each of those cells. So if you look first at the warm colors, the red and the orange control clones, what you can see is a certain fluorescent signal that indicates this purple diamond yellow circle disaccharide is on the cell surface properly. But when you delete GALE and those two clones colored in green and blue, you see probably a 50-fold decrease in their presence on the cell surface. This is a common log scale on the x-axis. So that indicated that the deletion of GALE we saw on the previous slide causes imbalances in nucleotide sugars, like for example, the yellow circle UDP galactose. And we think that's being translated into defects on the cell surface from those sugar chains that you saw in earlier cartoons. So then, to make sure that we were right about that, we collaborated with Lance Wells and Chelsea Desbian, his graduate student at the University of Georgia, and they used mass spectrometry to characterize the glycans that we can see on the cell surface from control versus GALE deleted cells. So this is first the example of O-linked glycans, the ones attached to hydroxyls on serines or threonines in the cell surface. What you can see, I think pretty readily, just by eyeballing it, is that there's many structures in the control clones, but in the GALE-deleted clones, there's very few. They're profoundly deficient in the normal structures that you would see on the cell surface. And what's more, you can probably tell just by looking that the yellow color is almost absent, or is absent in this slide, basically undetectable from the GALE-deleted cells. And this makes sense because the yellow shapes, the circles being galactose and the squares being GalNAc, those are the nucleotide sugars that GALE is responsible for balancing. So it makes sense that we would see this defect, this profound loss of these O-GalNAc and O-Gal glycans on the cell surface when we delete GALE. We also looked at N-glycans on asparagine residues. There are differences here too. They might be a little bit more subtle. There's still quite a number of structures in the Gale-deleted cells that you can detect, but you'll probably notice here too that the yellow circles and squares, some of which you can see depicted on the left for the control cells, are largely absent. So to conclude this section of the talk, I want to zoom in and talk about just one glycoprotein. So I just showed you that there's some global profound differences in the glycans that you see in the cell surface when you do not have Gale working properly. An obvious question from that would be, does this matter functionally? Are proteins okay when this happens to them? Do they just shrug it off if they don't have normal sugar chains? Or is their function impacted? So the one example that I'm going to show you today is a protein called FAS. This cartoon depicts the FAS pathway for those that are interested. The 10,000 foot view that you need to know about for FAS for this talk is that FAS is what's referred to as a death receptor. It is a protein that, when triggered, causes the cell to undergo apoptosis or die by suicide. So this is actually a programmed normal process by which the immune system, for example, can activate FAS with another molecule in another cell called FAS ligand, denoted FAS-L at the top right of this slide. And then if the cell is, for example, a tumor cell or infected with a virus, the immune system can instruct that cell to kill itself. And this is a good normal process in health and can also be dysregulated in disease. So FAS, as it turns out, is a glycoprotein. It has N-glycans on it. And it was known from other people's work previously that those are important for the function of FAS. So we wondered, since we're seeing profound defects in glycoproteins in the cell surface, might this impair FAS function? There was no reported connection between Gale and FAS, but given our mass spec data and our other data, we wondered whether FAS, the death receptor, function might be impaired in Gale-deleted cells. So as a first piece of evidence that this might be the case, Alex and Alicia looked at whether FAS is abnormally glycosylated in Gale-deleted cells. It turns out that it is. So again, looking at this Western blot here, hopefully you'll be able to see my arrow. These bands that I'm indicating on the left in the control lanes, two different clones of control cells A and B, these are FAS right here. This is the correct molecular weight for FAS. What you'll notice over in the lanes with the Gale-deleted cells is that FAS is migrating faster, it's smaller. What that means is that it is not glycosylated normally here in the minus lanes, this one and this one. What you see is hypoglycosylated FAS, meaning it does not have enough it doesn't have normal sugar chains, not enough individual sugars in the chain. However, we know that this is due to the loss of Gale, because if we feed the cells galactose to bypass the metabolic block of deleting Gale, now you can see that we were able to restore FAS to its predicted molecular weight from the glycan chains. So in other words, the loss of Gale causes FAS to be smaller than you would expect. And we know that that's due to the sugar chains that Gale supports, because when you bypass that block with galactose in the medium, now you can restore the size of FAS. We also know that this is due to N-glycosylation, because if you use an enzyme called PNGase F to cut off all of the N-glycans on FAS, now you can see everything return to normal. So what I'm illustrating here is on the left, we have protein, FAS protein from control cells or from Gale-deleted cells. And just like on the left of the slide, you can see that the Gale-deleted, the FAS from the Gale-deleted cells is smaller. And then on the right is the same protein samples, but treated with PNGase F to get rid of the N-glycans. And now you can see that all of the FAS protein from all the cells runs at the same molecular weight, it's the same size. So that tells you that these differences that we were seeing earlier are completely due to N-glycans. So is Gale necessary for the function of FAS then, since we see that its glycans are abnormal? Indeed it is. So we knew from other work by Susan Bellis at the University of Alabama that when FAS is hyperglycosylated, which can occur in some tumor types, then it is resistant to signals from the immune system, and apoptosis is blunted. This is used by tumor cells to protect themselves from being killed by FAS or FAS ligand. We're seeing hypoglycosylation, so up below normal levels of glycosylation on FAS, and we wondered therefore whether that would hypersensitize FAS to stimuli. Indeed, that's what happened. So here, Alex and Alicia did an experiment where they treated cells with increasing doses of FAS ligand, depicted on the bottom there, to induce the FAS on the cells that they're experimenting on. They did this with 2 control clones, which you can see at the top right, and 2 Gale-deleted clones in the bottom left. And what you can appreciate from the cell viability measurements they did, depicted on the y-axis, is that the Gale-deleted clones are hypersensitized, meaning it takes a smaller concentration of FAS ligand to kill them through FAS than the control clones are. So in other words, Gale is necessary for the normal glycosylation and function of FAS. If you get rid of Gale, as we've done experimentally in this case, now you have a FAS protein that is hypersensitized to its own ligand. And I think this is kind of interesting because we haven't just broken the protein. It's not that it's no longer working. Instead, it's working too well in a certain sense. It is hypersensitized to its own ligand, almost like a gain of function. So what we interpret this to mean is that Gale, and therefore nucleotide sugar metabolism, is required to link metabolism, the availability of sugars, to apoptosis or cell suicide. So it's a pretty intriguing result in that case. So just to summarize what I've told you from this part of the talk, we believe that Gale is essential for normal nucleotide sugar metabolism and glycoconjugate biosynthesis, glycoprotein biosynthesis in human cells. As a particular example of FAS illustrates, we think Gale supports the glycosylation of certain proteins. I told you about FAS today, but we found other examples. And we think it helps tune the sensitivity of cells through the glycoforms of FAS to apoptotic stimuli. And then finally, we think Gale and nucleotide sugar regulation in general might form a broad link that connects nutrient sensing, metabolism, and receptor signaling. So receptor being the general term for proteins on the cell surface like FAS and other abnormal glycosylations that we observed on other proteins, other cell surface receptors in the Gale-deleted cells. So at this point, I want to acknowledge the people that did the work. Here you're seeing photos of the fabulous people who are in my lab currently. If you want to learn more about what we do, this is the website here. Actually, the landing page for the website is under construction, but please visit and click on the links because they all work. Also Twitter, or feel free to reach out to me by email. Collaborators in this project, I mentioned Alex Broussard, former lab member who pioneered this project. Jenny and Tim in the lab also helped him, and our colleagues Zichang and So-Young at Duke. Also Lance Wells, who I mentioned, and Chelsea Desbian at the University of Georgia. And Jen Kohler and work I didn't have time to show you today. Jen Kohler at UT Southwestern in Texas, and Nicole Nishan also helped us with this project. Finally, always important to acknowledge the sources of funding. In particular, I want to thank the Mathers Foundation in the bottom right part of this slide. They have actually recently given us a grant to extend our work on Gale into an animal model. We'll be making knockout mice, conditional knockout mice of the Gale enzyme, and then looking at how its role in nucleotide sugar metabolism is important in various tissues. So with that, I'd like to move on to the second part of my talk, which is relatively brief and focused on allyship. And I wanted to use the title Addressing Equity and Inclusion as a Practicing Biologist, because, you know, as is probably amply clear from the first part of the talk, you know, I consider myself a cell biologist, not an expert in social science or anything like that, you know, full disclosure. But I think it's important to think for those of us who are practicing scientists at the bench or at the blackboard or at the computer, how we can advance equity and inclusion interests from our own roles as practicing scientists. So I'm really honored and humbled to receive this recognition and be here with you today. And I do want to keep this brief because the focus of this terrific event is properly on the Black queer STEM community. So I'll just offer some brief individual thoughts. So the first thing that I think of in considering allyship is that it should really be everybody's duty, because injustice or discrimination or exclusion is everyone's responsibility. So I think I'm white, and in thinking about, for example, anti-Black racism, it's important for all of us in the scientific enterprise to address injustice or racism, and certainly not let it fall disproportionately on those who are in historically excluded or marginalized groups, like, for example, Black folks or certain gender or sexual identities who have also been excluded and marginalized, to remedy these sources of exclusion or discrimination that they encounter. It's all of our responsibility, and that's what allyship is about. Many scientists, maybe you've run into this too, I found that many scientists believe that science or academia in general is a pure meritocracy. This is clearly not true. There's all sorts of studies and data indicating that humans are biased, explicitly biased often, implicitly biased always. There is discrimination in all its forms. It's pervasive. This happens at the personal level, so individual humans can be discriminatory and biased, but it also manifests in systems and structures, right, that occur in academia and science in general, and those might be more pernicious and more important in general. So this is clearly unfair when it harms certain groups based on their racial or ethnic or gender or sexual identities, ability versus disability. And this can occur in my own career, just to list some examples, in school admissions and job opportunities, professional advancement, pay, research funding like NIH grants. Therefore, you can see that individual scientists who are from historically excluded groups are harmed. This can also hurt science in a larger sense, in that it can exacerbate scientific problems like understudied or neglected topic choices, health disparities, to use an example in biomedicine, my own field, which can disproportionately affect people of color or the LGBTQ community, for example. So not only are this lack of meritocracy and this sort of pernicious discrimination affecting individuals' careers, which is bad enough, but also it can affect public health and the scientific enterprise itself. So for all of these reasons, it should be our collective responsibility to engage as allies to abolish this. I would also say for people who are skeptical of this in the academy that, in my view, equity and inclusion are needed for true inclusive excellence, really anywhere, but I'll speak specifically about academic biology. When they're absent, it's a threat to academic freedom. So what do I mean by that? I think inclusive excellence in biomedicine and biological sciences, probably all scholarly fields, requires that people like graduate students, postdocs, faculty, requires that all of us be comfortable being our whole selves and being able to bring our various identities to our work and our study. So this enables the pursuit of science by making people comfortable, knowing that they belong, enabling them to persist in the field when they go from junior colleagues to more senior in their field. And also just to speak up with ideas, suggestions, new scientific theories. That's what academic freedom is, and an environment where equity is lacking, discrimination occurs, people feel uncomfortable, they don't belong. There's a chilling effect on the interchange of knowledge and the pursuit of science. So for that reason as well, this is a subject for all of us to be concerned about, and allyship should address itself to this threat to academic freedom. This is something that we have to address, all of us where we are. Sometimes I'll hear colleagues in the faculty, if you're talking about a lack of representation from this group or that group, or a structural inequality, they'll blame the pipeline, or they'll say, it's the K through 12 education where it happens. It's too late when it gets to us. I reject that kind of thinking. It's not to say that there aren't problems elsewhere in the country that contribute to this, but I think it would be a cop-out for us to not do what we can where we are. We know that there are inequalities to address in our own local environments, and it's incumbent on us to do that. I would also say it's important to use an intersectional lens in looking at these questions and problems. This is certainly appropriate for a Black and Queer STEM town hall to discuss intersectionality briefly. I think it's probably no news to this audience that the gay rights movement, if you can call it that, or the queer community in general, has had a racism problem, anti-Black racism and other racist problems in its past and in its present. That's just the same as the country that we're in. Analogous situations for women, trans folks, gender non-binary folks. And this is something that you have to acknowledge and tackle head on and not ignore. And that's in one way thinking intersectionally about these groups and how the experiences of people will differ based on their belonging to one, two, three, many of these groups together. So I think we as working scientists have to bear that in mind when we're trying to create an equitable and welcoming environment in our labs, in our universities, and our institutions. So thank you for having events like this today that highlight the intersection of some of these particular challenges for these groups. I also want to say that people with power and privilege, like myself, I got tenure in 2019, very happy about that, are particularly well positioned for, and I would say particularly called to, allyship, or we should be. It's important for people like me who have responsibility and power to leverage that to create or expand the sort of belonging and equity that we're talking about in academia. So, bumper stickers or hashtags or emojis in your Twitter bio, these are good symbols. These are good things to do. Symbols do matter, don't get me wrong, but they're not enough. If they exist in the absence of action, then they're sort of performative gestures. And I think it's, to me, more important, maybe more interesting to think about how we can leverage actual power and money to change systems and structures and make material improvements in people's lives. And that is something that allyship should address itself to. Last general observation I would make is that allies can learn ourselves and take action while remaining mindful of our own lived experiences. So I think some scientists from well-represented groups, white folks, let's say, may have vague good intentions and they want to promote equity in their own institutions, but they don't know how. They might be afraid of saying the wrong thing. They might be afraid that they don't know what they're doing. But concrete action is still necessary and still key. If we want to fix injustices and create equitable systems and structures, it's not enough for individuals to just be not racist or be not homophobic. You have to get educated and then take action when it's necessary. So I'd like to use this example I heard from David Asai, who's the Senior Director of Science Education at the Howard Hughes Medical Institute. Hi, David, if you're listening. I saw him give a talk once where he sort of made an analogy for earlier in his scientific career. He was a biologist running a lab for many years. And at one point earlier in his scientific career, he found, if I understood his anecdote correctly, that he needed to learn how to make some monoclonal antibodies for his lab to use as tools to study a particular protein. And he had never made monoclonal antibodies before, and this was going to be a completely new thing that he had to learn as a biologist, but he did it because it was necessary for his science in order to fulfill his mission to run his lab the way he wanted to. He needed to learn this particular skill, and so he went out and did it. And I think that's a good analogy to use for colleagues who are maybe shy about allyship and say they don't know, or they're afraid of saying the wrong thing. That's okay, but you can learn. And if something is important for you fulfilling your role as a faculty member or a mentor, for example, an administrator, then it's important to get off the couch and take action, educate yourself in the way that you need to, and then don't stop there, but continue to play an active role in the effort to expand equity. Of course, when we're doing this, speaking as an ally myself, I'm always mindful of trying to maintain a certain epistemic humility. So I do not presume to know what's best for other people. I do not have the lived experience of being, for example, a racial or ethnic minority in the United States. And I never want to pretend or forget that I don't have those perspectives. So I'm not saying that. I'm saying that it's important to learn and take action as an ally in an appropriate way that remains mindful of our own lived experience and what we share and what we don't share with others in that regard. So no matter who we are, I think we have a role to play as a mentor, as a PI, as a member of an admissions or job search committee, et cetera. And it does not let us off the hook just because there are things that we need to learn in order to do those jobs more equitably and inclusively. How does this tie in more specifically with the topic of today? I would say one idea that I have is that I think being out in STEM can advance equity. I'm gay. I have a partner. This is a picture of us actually from a university-wide email blast that Duke did a couple years ago, where they wanted to find for Valentine's Day couples who both work at Duke. It's kind of a cheesy thing. And we agreed to get photographed and get our cheesy photos sent out to the entire university. I would like to think, I hope, that having visibility in this sort of way that looks good on a PowerPoint slide maybe, or not good, but it's on a PowerPoint slide. But other things, like working with the OSTEM chapter on campus, for example, sort of being out in a STEM field, I would hope would help to advance equity. I think that this is important for people serving as individual role models or individual allies, individual sources of support or mentors. But I think in the long term, it can also help change the culture. I saw a talk by Taeshik Yoon, who's a chemist at the University of Wisconsin once, and he made the point that for decades, social scientists have been asking elementary school children to draw a scientist. They give them crayons and paper and they say, draw a scientist. And what you can see is most children draw men, or in this case, this is analyzed through a gender binary lens, but primarily they're drawing men. But over time that's changed, and more children over time have drawn more and more women when they're asked to draw scientists. One way of thinking about this is that you're not talking about specific individual female scientists, you're talking about children's general idea of who a scientist is or could be. So I think if you are out in STEM, for example, or showing representation, it matters not just to provide individual examples of specific scientists, but it can change the culture eventually. It changes the water we swim in. And hopefully that sort of representation can help people, more junior colleagues in the same field. Last thing I'm gonna mention is my work for the last 5 years with the American Society for Cell Biology, which is a professional society in my field, with the Minorities Affairs Committee. So this is a group that promotes the science and professional development of historically excluded racial and ethnic group scientists. The MAC runs a large number of programs on grant writing, professional development, travel awards. We promote cross-cultural collaborations, publish our interventions in peer-reviewed journals. I'd encourage you to check us out at this website below. Importantly, in my mind, we write federal grants and get money from the National Science Foundation, from the NIH to operate these programs. In 2019, the last year I have data handy for, our operating budget just for our committee was about $770,000. This is leveraging real resources to promote the inclusion and professional advancement of folks in these communities in cell biology, and I'm very proud of that. For the last couple of years, I've been co-chair of the committee with Professor LaTanya Hammonds-Odie from Georgia Gwinnett College. That was, in a way, sort of a stretch or a challenge to allyship to be able to navigate that space and contribute to this effort, while also remaining, as I mentioned earlier, aware of my sort of lack of lived experience as a historically excluded racial or ethnic group in cell biology. And I hope that through careful listening and my hard work and cooperation with my colleagues on the MAC, that has been a positive thing for me and been able to contribute in a meaningful way to that effort as well. And then finally, I'll mention the Mosaic program at ASCB that we're starting. We're very excited about this. This began just this year with a $1.3 million grant from the National Institutes of Health. That's going to last 5 years. It's going to allow us to create a cohort-based program for postdoctoral scholars who are from historically excluded racial and ethnic groups in cell biology and allied fields. And there's going to be sort of supplemental mentoring and professional development that ASCB runs for postdocs from all over the country. The end goal of this program is to diversify tenure track faculty at research universities. And a second important goal is to affect cultural or structural change to those schools. So not just get jobs and professional success for the individual fellows in our cohorts, but also to help the universities where those postdocs work to do a self-reflective process and become more equitable themselves. So we're not focusing only on individual scientists here, but we're trying to facilitate culture change that will improve the climates, the cultures at universities, research universities across the country, hopefully to the benefit of many scientists in many departments in the future. So we want K99R00 scholars to be part of the ASCB Mosaic program. So if you're a bioscientist, please check that out. You can also Google NIH Mosaic program for more information on the NIH side of things. And we also need mentors. So if you're a faculty member, for example, who might be interested in serving as a supplemental mentor for a fellow in this program, I would love to hear from you later on. And with that, I will conclude. I just want to thank the organizers again for this great opportunity. I'm really honored to have been a part of this today, and I look forward to interacting with all of you, hopefully off of Zoom in person sometime soon in 2021. Thank you.
Boyce, thank you very much for your allyship. We need more individuals in science like you that are not only willing to give a talk for research, but also to talk about the differences that lie between each of us as scientists, but how we can embrace them and make a better culture. And we also want to say thank you for your work as one of the co-directors of the American Society of Cell Biology's MAC committee. You're doing a phenomenal work, and congratulations again on the UE5 from NIGMS. I think that Mosaic grant is going to do fabulous wonders for the American Society of Cell Biology's role in playing in how faculty can be mentored and developed into scholars and seen as respectable, but also high achieving scientists in multiple fields, but most importantly in cell biology. And I just want to say I'm inspired by what you do, and we really appreciate all the things that you're doing, and congratulations again on your allyship award. Our next speaker is Dr. Whitney Gibbs. Dr. Whitney Gibbs received her PhD from Medical University of Southern Carolina and Dr. Rick G. Shillman's lab, and she is now a postdoctoral fellow in Dr. Matt Lavoie's lab at Harvard University and Boston Children's Hospital. Please welcome Dr. Whitney Gibbs.
Good morning. My name is Whitney Gibbs, and I'm currently a postdoctoral fellow at Boston Children's Hospital and Harvard Medical School. Today, I'm going to give you a snapshot of the work I performed as a graduate student in the laboratory of Rick Schnellman at the Medical University of South Carolina. There, I studied the role of the 5-hydroxytryptamine 1F receptor in mitochondrial biogenesis and acute kidney injury. As you can tell from our title, our lab focuses on the mitochondria. The mitochondria is a double membrane organelle, and we probably all know it as the powerhouse of the cell as it generates energy through the form of ATP. Mitochondria is also involved in a number of cellular processes, such as in the regulation of apoptosis or cell death. It functions as a calcium scavenger, and it also regulates the levels of reactive oxygen species production under both physiological and pathological conditions. As the mitochondria regulate a number of cellular processes, it is no surprise that mitochondrial dysfunction or dysregulation of mitochondrial homeostasis has severe pathological consequences. Injury to the mitochondria following toxicant exposure or direct injury, such as in the form of ischemia, reperfusion, all contribute to deficient mitochondrial function. Mitochondrial stress or increased free radical production, disruption in electron transfer chain activity, and diminished ATP production all disrupt a number of cellular processes, such as cellular repair and proliferation. It increases cellular death. And it's been well documented that mitochondrial dysfunction and altered mitochondrial homeostasis leads to the propagation of injury and the subsequent tissue and organ dysfunction. And this is primarily relevant in organs that have high metabolic demand, such as the brain, the liver, the heart, and the kidney. Diseases such as Alzheimer's disease, Parkinson's disease, acute injuries such as myocardiac infarction, as well as acute kidney injury, have all been characterized by persistent mitochondrial dysfunction. And so our lab is primarily interested in understanding how we can restore mitochondrial function as a potential way to reduce the propagation of injury in these acute and chronic disease states. And we particularly studied mitochondrial biogenesis as a way to do this. So what is mitochondrial biogenesis? Mitochondrial biogenesis is defined as the generation of new functional mitochondria. New mitochondria is formed from existing mitochondria. A new mitochondria buds from existing mitochondria and functions just as any normal mitochondria. This is a very complex process as it requires the coordination of both the nuclear and mitochondrial genomes. The mitochondria has its own DNA, which encodes for about 13 mitochondrial proteins. And so in order for mitochondrial biogenesis to happen, it requires the communication between the nucleus and the mitochondria. This is all coordinated by a protein called PGC1 alpha, which is termed the master regulator of mitochondrial biogenesis. There's a number of pathways that activate PGC1 alpha, such as if the cell senses low ATP levels, PGC1 alpha will then be stimulated. PGC1 alpha stimulation leads to increase in mitochondrial DNA transcription and protein translation, leading to mitochondrial biogenesis. So as I mentioned earlier, our lab is particularly interested in acute kidney injury, or AKI. AKI, very briefly, is defined as an abrupt decline in renal function following some insult. These insults are classified in three ways: pre-renal, disruption in blood flow going to the kidney, such as after a cardiac event; intrarenal, which is defined as direct insult to the kidney; and post-renal, downstream of the kidney, such as like a bladder infection or bladder disruption, can also lead to abrupt decline in kidney function. And unfortunately, 50% of the patients that do not recover from AKI unfortunately die. And this is because there's no cure for AKI. The treatments are limited to supportive measures such as dialysis. So our lab generated a mouse model of IR-induced AKI, or ischemia reperfusion-induced acute kidney injury. And about 10 years ago, the graduate student at the time wanted to look at not only acute injury in mice, but also look to see how proteins involved in electron transport chain activity correlates to acute kidney injury. So first, he prevented blood flow to both kidneys by clamping the renal pinnacles to both the left and right kidney. In doing this, he prevents blood flow for about 20 minutes. And after 20 minutes, he releases both clamps, allowing for blood flow to return to the kidney or reperfusion. Then he follows the mice for about 7 days post-surgery. And at 24 hours, mice have maximal renal dysfunction. And this is measured by serum creatinine. So serum creatinine at 24 hours is significantly increased. And as mice have been followed 7 days post-surgery, while renal function does become better, it never returns to that of sham animals or the no surgery animals. So these mice have persistent renal dysfunction. He also looked at a number of mitochondrial proteins, but I'm specifically showing you some of the proteins that are involved in electron transport chain activity. ATP synthase beta, COX-1, and NDU-FB8 all are components of the electron transport chain. And at about 72 hours to 144 hours, or 3 to 7 days post-surgery, these protein levels are substantially decreased and persistently decreased, demonstrating that mitochondrial dysfunction or dysregulation of mitochondrial homeostasis is altered in mice that have been subjected to renal injury. And this also is associated to prolonged renal injury, as well as changes in renal tubular morphology, where these cells become very necrotic and do not recover 100% post-surgery. And so our lab was, at the time, developed a high throughput screening to assess a number of chemicals, small molecules, to determine if they're able to induce mitochondrial biogenesis, hopefully to rescue the deficiencies that we see in mitochondrial protein levels following AKI. And so to do this, we isolated primary renal proximal tubule cells from rabbits, and we plated them in 96-well plates. And these cells are plated in low glucose, high oxidative phosphorylation conditions. So we know that the mitochondria are working at normal rates. We then exposed these cells to a chemical library, a variety of small molecules. And then we used the seahorse to analyze oxygen consumption rates. And oxygen consumption is a surrogate marker of looking at ATP or mitochondrial function, because as the mitochondria consume oxygen, they produce ATP. And so we looked specifically for compounds that increase oxygen consumption rate or increase mitochondrial function. And the best part about this assay, outside of the fact that it's so robust, it also is measuring mitochondrial function in live cells. The chemicals that we identified in this screening were then validated through a variety of different measures, including electron transport chain, mRNA and protein levels, mitochondrial DNA copy number, which is a surrogate of looking at the number of mitochondria that are produced in the cell, as well as other methods like ATP levels. So one of the most robust chemicals that we tested were inducers of the 5-hydroxytryptamine 1F receptor, or the 5-HT1F receptor, in two compounds, LY334370 and LY344864, are both agonists of the 5-HT1F receptor, increase oxygen consumption rates in the renal proximal tubule cells at nanomolar concentrations, so very potent inducers of 5-HT1F receptor. We also did this alongside methylated 5-HT or serotonin, and that also increased oxygen consumption rates. So these data definitely suggest that stimulation of the 5-HT1F receptor increases mitochondrial biogenesis through the measurement of oxygen consumption rates. We then took the renal proximal tubule cells and treated them with LY. We moved forward with one of the compounds, LY344864, which I will call LY because it's very long. And we looked at ATP synthase beta, COX-1, and NUFB 24 hours after treatment of the renal proximal tubule cells. And again, we see increase in these protein levels, demonstrating that the 5-HT1F receptor is somehow mediating mitochondrial protein levels as well as oxygen consumption rates of the mitochondria. And so this data was particularly exciting because this was the first time that the 5-HT1F receptor was linked to mitochondrial biogenesis. And so the graduate student at the time wanted to ensure that these compounds was directly hitting the 5-HT1F receptor. And so she used siRNA technology at the time to knock down the 5-HT1F receptor and then treat it with LY. And she was not able to see that increase in electron transport chain protein expression as she did in the wild type cells. So this experiment told us two things. One, our compound is selectively targeting the 5-HT1F receptor, and stimulation of the 5-HT1F receptor is directly responsible for this LY-induced increase in electron transport chain expression. So that's great. The 5-HT1F stimulation increases mitochondrial protein expression, but what about when it comes down to pathological conditions and acute kidney injury? So she took mice and she again performed IR-induced AKI surgery on the mice. 24 hours after surgery, we see this robust decrease in renal function. And again, in the wild-type mice, they begin to recover a little bit, but not nearly as much as we need to see for substantial recovery of renal function. However, when she started treating with LY at 24 hours after surgery, and she treated daily for 6 days, she observed the acceleration of recovery of renal function in these mice. And they're pretty much no different than mice that did not receive any surgery. So stimulation of the 5-HT1F receptor after surgery, so this is post-surgery, not pre-treatment, increased or accelerated renal recovery in these mice. She also looked at kidney injury molecule 1, which is only expressed when the tubular cells of the kidney are injured. So this is tubular-specific marker that's only expressed following injury. And so the no surgery and sham animals do not have KIM1 expression. However, at 6 or 7 days post-surgery, we have the substantial expression of KIM1. However, when mice were treated for 6 days with LY, while KIM1 protein levels are still there, it is reduced by about 40 to 50%. So demonstrating that kidney injury, tubular injury, is not as elevated in when you treat out with LY or stimulate the 5-HT1F receptor. So how does this correlate to mitochondrial function or biogenesis? And so we looked at mitochondrial DNA copy number, again, as a surrogate number of mitochondrial biogenesis. And mice that were undergo IR surgery that were treated with just vehicle or saline had about a 50% decrease in copy number about 6 days after surgery. However, after treating with 6 days with LY, these mice are beginning to recover their mitochondrial DNA copy number, again associating mitochondrial dysfunction, or excuse me, mitochondrial homeostasis recovery with the recovery of renal function in mice that were treated with LY. And so this again was very exciting data that we were able to link this novel target of mitochondrial biogenesis to a recovery of an acute injury. So when I came into the lab, the question was, what is the mechanistic link between this novel target 5-HT1F receptor 2 mitochondrial biogenesis. And so again, I'm reminding you, there's a number of pathways that lead to a stimulation of PGCL1 alpha and mitochondrial biogenesis. In the interest of time, I'm not going to show you all the data that I did to figure out which pathway was responsible, but I will go ahead and tell you that the nitric oxide cyclic GMP was actually elucidated as the major pathway linking 5-HT1F receptor to mitochondrial biogenesis and PGC1 alpha. And there's a number of studies out there supporting this pathway as a mechanism that leads to PGC1 alpha activation in mitochondrial biogenesis. And so very briefly, I performed the same assay, seahorse assay, as previous students in the lab had done, where I treated venoproximal tubule cells with LY for 24 hours and was able to show increased oxygen consumption as well as mitochondrial DNA copy number after 24 hours with LY. However, when I used inhibitors of G beta gamma, so the 5-HT1 alpha is a GPCR, and so when I inhibit G beta gamma activity, and then also components of the nitric oxide cyclic GMP pathway, AKT, nitric oxide synthase, GMP, cyclic GMP, and PKG, inhibitors of all of these components prevented this LY-induced increase in oxygen consumption and mitochondrial DNA copy number. Again, providing substantial evidence that this pathway is indeed mediating 5-HT1F-induced mitochondrial biogenesis. So I did a time course where I needed to show directly that LY was increasing these components. So about 15 to 30 minutes following LY treatment, AKT phosphorylation was elevated, and this was a direct result of G beta gamma activity or activation of this GPCR. When I block G beta gamma activity, I reduce or prevent AKT phosphorylation. I then looked at eNOS phosphorylation, and an hour and 2 hours post-treatment, we see an increase in eNOS phosphorylation. And again, this is directly downstream of AKT and G beta gamma, as inhibitors of both of these prevented eNOS phosphorylation increase. So again, LY is acting on this 5-HT1F receptor, which is a GPCR, and we increase AKT phosphorylation as well as eNOS phosphorylation. The next step of this pathway I looked at is cyclic GMP, and I see an increase in cyclic GMP following treatment. And then also phospho-VAS, which is a surrogate marker of PKG, was also increased. And this was again downstream of cyclic GMP as well as PKG itself. So treatment with LY, we activate AKT phosphorylation, eNOS phosphorylation, as well as cyclic GMP levels, which I show in some other data a little bit more elegantly that PKG is also activated. PKG has also been shown to directly act or indirectly lead to processes that activate PGC1 alpha phosphorylation. PGC1 alpha undergoes a number of post-translational modifications, one being phosphorylation. And PGC1 alpha phosphorylation at serine-threonine sites has been shown to activate PGC1 alpha. And so at the time, PGC1 alpha antibodies were pretty bad. Western blot analysis with them were tight, they were hard to get, and so definitely immunoprecipitation was out of the question. So what I did was, unfortunately, I was limited to looking at it from the opposite way. I pulled down phosphorylated serine-threonine from proximal tubule cells, and then I bought it for PGC1 alpha. And after LY treatment, you see this increase in phosphoserine-threonine phosphorylation of PGC1 alpha. And this looks to be dependent on AKT as well as PKG, because when I inhibit both of these, AKT and PKG, I prevent this increase in PGC1 alpha phosphorylation after LY treatment. So very quickly, I know I flew through this in the interest of time, but LY is activating the AKT eNOS cyclic GMP pathway to stimulate PGC1 alpha phosphorylation, which has been shown to then translocate into the nucleus to activate transcription of proteins such as those involved in electron transport chain activity. At the time, a graduate student in the lab was also looking at ERK phosphorylation, and decrease in ERK phosphorylation he showed to be linked to mitochondrial biogenesis. And so I then also looked at ERK phosphorylation, and I showed that LY actually reduces ERK phosphorylation about an hour after treatment. And this also looked to be dependent on AKT because when I blocked the AKT inhibitor, ERK phosphorylation was reduced. And so I expanded this work, again, not showing you a lot of it in the interest of time, but I also was very excited to link two distinct pathways to be downstream of LY stimulating PGC1 alpha or mitochondrial biogenesis. So the pathway shows you eNOS and PKG activating PGC1 alpha phosphorylation also seem to contribute to ERK dephosphorylation of ERK through AKT. Also, saw increase in FOXO3A, which is a transcription factor known to activate PGC1 alpha. So for the first time, I linked 5-HT1F receptor to mitochondrial biogenesis, not through one pathway, but two pathways that were working simultaneously. And so this took about the first 1.5 years, 2 years of my graduate work. And so I was ready to get into animals and understand how the 5-HT1F receptor is directly mediating mitochondrial homeostasis as well as renal function. And so to do this, I generated a 5-HT1F knockout mouse. Of course, there were none at the time. So I generated a 5-HT1F knockout mouse colony. And here I'm showing you that there were no transcript levels of the 5-HT1F receptor in a number of tissues in the mice. And everyone has a downside of their graduate work, and this is mine. I was hoping to get really sick mice that had this aberrant physiological dysfunction, but it didn't happen. All these mice were normal, healthy, viable mice, and I looked at a variety of different parameters and didn't see any changes. And like I said, everyone has that low point. This was my low point. And so I also then was like, okay, let's look at renal function. Let's look at renal biology. And these mice had normal renal function. I looked at mice as they aged. I'm not showing all the data, but their renal function and their tubular, they had no tubular injury. Their morphology of tubular cells were all fine in these mice. So then I'm like, let's look at pathological conditions. How does the 5-HT1F receptor contribute to renal recovery following injury? Because I already knew that if we treat with LY, if we stimulate 5-HT1F receptor, we accelerate recovery. So how is the 5-HT1F receptor modulating this? And so I did the same experiment or surgery that previous students in the lab had done, where I induced ischemia reperfusion, induced AKI. And at 24 hours, I looked at both the wild type and knockout mice. And at 24 hours, both had increased renal dysfunction as expected, but there was no difference in renal dysfunction in the wild type or knockout mice. Then I looked at the serum creatinine and blood urea nitrogen levels. I also looked at tubular injury. So again, KIM1 is a protein that's only expressed in the tubular cells and only expressed under injury. NGAL is another injury marker that's increased during injury as well. So while at 24 hours there was no difference in renal function, tubular injury was definitely potentiated in the knockout mice, as I'm showing you here with both KIM1 and NGAL protein expression being elevated. So at 144 hours after surgery, so 7 days post-surgery, I also looked at renal function in the knockouts compared to wild types. So again, at 24 hours, we get the substantial increase in or decrease in renal function as demonstrated by increased serum creatinine and blood urea nitrogen levels. The wild type mice, which is the solid line, they begin to recover a little bit, but not a lot, which has been shown again in the previous study. So the same type of injury is happening in these mice where not much recovery is happening, but to some degree. The knockout mice, which is the dashed line, the top dashed line, we see that these mice don't recover at all. While the wild type mice make some level of recovery, these mice do not recover at all at 144 hours. These knockout mice are pretty much staying pretty consistent with the level of injury as we saw at 24 hours. And again, this was shown through renal tubular injury. At 24 hours, they had increased tubular injury as shown by KIM1 and NGAL, and this persisted at 7 days. So the tubular injury is consistent and sustained. So this is exciting because now I'm seeing direct evidence that the 5-HT1F receptor is required to have proper recovery of renal function and tubular function after AKI. So how does mitochondria play into this? My claim to fame with my project is that the 5-HT1F receptor is modulating mitochondrial biogenesis, and this is the mechanism that is responsible, or at least plays a large part in rescuing renal injury. So I looked at a variety of markers of mitochondrial biogenesis, one being PGC1 alpha, as well as electron transfer chain proteins, NUSF1, COX1, and TFAM. And at 24 hours, we see decrease in PGC1 alpha in the knockout mice, as well as TFAM, which is a transcription factor in the knockout mice, whereas the wild type mice at 24 hours, we don't see any true mitochondrial protein level changes, which again was supported in the initial model that was done by a previous graduate student. At 144 hours, though, we see that in the wild type mice, we do see some disruption in electron transport chain proteins. However, in the knockout mice, we see sustained decrease in PGC1 alpha, NUSF1, electron transport chain proteins that is consistent with this delayed renal dysfunction that we see in the mice and the knockout mice. So both renal dysfunction, mitochondrial protein levels are decreased. More likely, mitochondrial biogenesis is disrupted in these mice by looking at PGC1 alpha and some of these other proteins that are required for proper mitochondrial function, as well as ATP levels. So I also looked at ATP levels in these mice. And at 24 hours, which is no surprise, both the wild type and knockout mice have decreased ATP levels about 50% compared to those wild type controls. However, at 144 hours, ATP levels or energy pathways are beginning to come back up in the wild type mice again, which kind of correlates to what we see in terms of renal injury. They're starting to recover, but they're not there yet. However, in the knockout mice, ATP levels remain persistently decreased in the knockout mice at 144 hours post-injury. And so this definitely goes along with decreased PGC1 alpha protein expressions, electron transport chain protein expression, and decreased ATP levels. And so these mice, while they didn't have any physiological deficiencies in terms of mitochondrial levels as well as renal function, under IR-induced AKI, the knockout mice have increased tubular injury, suppressed mitochondrial protein levels, and decreased ATP levels. And I also showed in some other studies that they have elevated oxidative stress. And so this tells us that the 5-HT1F receptors plays a protective role in IR-induced AKI, or protective role in renal function, and this protection is at least partly mediated through regulating mitochondrial protein levels and mitochondrial homeostasis. And so in summary, a large part of my graduate work, I was able to elucidate a novel mitochondrial biogenesis pathway where the 5-HT1F receptor is inducing two mechanisms simultaneously by increasing the AKT, ENOS, PGC1 alpha pathway, while simultaneously decreasing the ERK, phospho3A, which both activate PGC1 alpha. Again, the 5-HT1F knockout mice are healthy and viable, but when we stress them, they have increased tubular injury and persistent renal dysfunction following injury, and we also were able to correlate this with persistent reduction in mitochondrial biogenesis and ATP. And so overall, the overall finding of my graduate work is that the 5-HT1F receptor not only induces mitochondrial biogenesis through a novel transduction pathway. This is the only pathway to our knowledge that has been elucidated to show two pathways simultaneously activating PGC1 alpha. And this could also contribute to the robustness of the LY compounds in inducing mitochondrial biogenesis. And this protects the kidney from any further or potentiation of renal dysfunction following AKI injury. And so this work was obviously done in Schaumann Lab, and there was a variety of different people involved in the project, support and advice from the Schaumann Lab members, as well as collaborators, and obviously funding. My graduate work started in Charleston, South Carolina, and then about 3 years in, my PI moved to the desert, to Tucson, Arizona, so I completed my work in Arizona. So this project had a lot of ventures to it on the science side as well as location. And so I hope you guys enjoyed and learned something new about mitochondrial biogenesis and acute organ injuries. And if you have any questions, I will be around later for questions. Thank you so much. Stay safe.
All right, thank you so much, Dr. Gibbs. That was an amazing talk. Our last speaker of the day is Dr. Derek Applewhite. He received his PhD in cell and developmental biology at Northwestern University in Dr. Gary Borisi's lab. Dr. Applewhite then went on to do a postdoctoral fellowship at the University of North Carolina, Chapel Hill, studying the coordination between actin and microtubules and their cross-linking proteins. Dr. Applewhite has also been an advocate for undergraduate mentoring and education, and he's an advocate for equity in both the Black and LGBT community by playing a role in the American Society of Cell Biology's Minority Affairs Committee and also their LGBTQ+ task force. He's also here to tell us about his journey in STEM and how to make everyone have a seat at the table. Please welcome Dr. Derek Applewhite, and congratulations on the Journey Award.
Good afternoon, everyone. Thank you for this opportunity. My name is Derek Applewhite. I'm an associate professor of biology at Reed College, and today I'm going to share with you the work from my lab. So the title of my scientific talk is The Role of Split Discs in the Regulation of Non-Muscle Myosin II Contractility. Now I know that's a mouthful, so hopefully I have a chance to break this down so that you can understand exactly what it is that we're doing in the lab. And this is a beautiful picture of Reed College, located in Portland, Oregon. In springtime, so you can kind of see the cherry blossoms at their bloom. So there are two parts to this talk: the scientific part, which I've already alluded to the title, and then the second part is going to be my journey as a scientist. And I have an aspirational portion to this, which is what can we do to make sure that everyone gets a seat at the table? So stay tuned for that. That'll come after my scientific talk. So the first part, the role of split discs and the regulation of non-muscle myosin II contractility. And before I get too deep into the weeds, I want to introduce those who actually have done the work. I am but their mouthpiece. Aidan Turan is a current post back in the lab. He was a senior last year, and a lot of this work was his thesis for his senior year. JJ is also a research assistant now, and he's been in the lab for about 2 years. Amy Plantenkamp just left. She left for graduate school at the University of Washington, and Sushruta is a current senior at Reed College. He's actually a physics major, a biophysics major, so there's some biophysics aspects to the research that I'll be sharing with you today. So non-muscle myosin II is a molecular motor protein, and molecular motor proteins at the very basic take chemical energy and turn them into mechanical work. So there are three main families of molecular motor proteins. Dynein, and here what we're seeing is the cilia of a tetrahymena, and dynein is pulling on this microtubule-based structure so that this organism can swim through a pond. Kinesin, which is another microtubule-based motor, kinesin is responsible for the motility that you're seeing here. This is a S2 cell, it's a Drosophila fly cell type of cell line that I work with. And the bright white structures that you see moving back and forth are peroxisomes. And so kinesin is responsible much for the motility that you're seeing here in these peroxisomes. And then the final family of molecular motor proteins are myosins. This is a cardiomyocyte, so a pre-heart cell on a soft substrate. So spontaneously cardiomyocytes can pulsate like this, much like your heart pulsates. And so what we're watching is the activity of that myosin pulling on the cardiomyocyte, and then in turn seeing it take this shape change happening. Let's dive a little bit deeper into myosin, since that's actually the target of this talk, the subject of this talk. Myosin is an actin-based motor. So we have microtubule-based motors, which are dynein and kinesin, and then we have actin-based motors, which are myosin. And myosin II is just one of many different myosins. But here we're looking at a cell undergoing cell division, something that biologists and non-biologists alike are familiar with, at least through pop culture. This cell is just dividing into two cells. But what may not be clear to you is in order for the cell to divide into two, we have to have constriction. We have to constrict. And so myosin is this actin-based motor that drives this constriction, allowing the cells to become two different cells. Myosin is also really important for development. And so what we're looking here is a fly cell undergoing development. And so we're having an invagination or an internalization of a layer of cells, because at the point of this development, there was just one layer, one ball of cells. But of course, Animals, organisms, even flies are made up of different layers of tissue. So the invagination that you see happening here is driven again by myosin during a process called morphogenesis, which is just simply cell shape change. So as myosin pulls on these cells, it causes part of the cell to shrink, but if all the cells sort of shrink at the same place, it actually causes the cells to be driven inside of the organism, allowing for a new tissue level to be formed. Myosin's also really important for cell migration. So here we have in bright white again, we see this sort of spontaneous sort of almost celestial image of myosin sort of oligomerizing into these complexes that again are contracting, so allowing the cell to migrate. So if we were to watch the cell for a lot longer than the time lapse of this movie, we would see the cell actually crawling, because cells crawl for many different reasons. And myosin is really critical for cell motility. At its very basic level, myosin is regulated by a phosphorylation event, a post-translational modification that happens to the cyan colored or the little light blue colored blobs on this image. They're phosphorylated, and this phosphorylation, this post-translational modification, causes the molecule to open up. So it's normally in a closed conformation that does not interact with actin, so it cannot actually do its job. It needs to be modified by this phosphorylation to open up the molecule. But even then, it's a really terrible motor protein. It actually has to work in teams. So it actually oligomerizes into higher ordered structures that then bind to actin, and then which is in red here. And then as myosin walks along the actin filaments, it actually causes those filaments to slide. And that sliding causes contractility that you saw in those three examples I showed you previously. And so all myosin is doing is walking along actin and causing the actin to slide across one another. That actin, of course, is connected to other things. So when the actin gets pulled, it pulls those other things with it, causing cell shape change that we saw during cytokinesis or the cell division, or during morphogenesis of that fly embryo, or when a cell is trying to migrate. So that is at the basis of how myosin works. And if we were to take a look at this a little bit more closely, again, this is Drosophila. This is a fruit fly cell. I study fruit fly cells, but a lot of the proteins that are important for fruit fly cells are really important for human cells. So they're a really great model to understand what's happening at the human level. So what we're looking at again is actin in red, and spaghetti squash, which is a component of non-muscle myosin 2. Flies have really bizarre cutesy names at times, so please forgive me. I'll try not to go back and forth between them, but spaghetti squash is just a marker for non-muscle myosin 2, our motor protein of interest. And then we have a merge of the two channels together here. So we're watching the cell in interphase, and we're seeing the formation of that higher molecular myosin oligomer that I just talked about in the previous slide. And if we were to zoom in a little bit more closely, we can see that, I like to imagine what's happening here is that you have non-muscle myosin 2 in its closed conformation, and then it's being activated by that post-translational modification, that phosphorylation, that allows it to incorporate into this larger structure, which then becomes contractile. So that is how myosin is sort of regulated in a nutshell. And so, a lot of time people give these scientific talks with this very specific hypothesis, and then the whole point of the talk is to sort of derive how we came to this hypothesis and sort of test this hypothesis. And I wanted to say that that's not always how science is done. So I kind of sort of themed this talk around the power of scientific exploration, or what really happens is, that looked funny, which is kind of how a lot of science is done, is you see something happening, you're like, well, that's funny, we should figure out how that's happening. And so using a bioinformatic query, which is just basically a blast search, I identified previously uncharacterized Drosophila proteins that have a specific domain. The domain happens to be a calponin homology domain. This is just a portion of a protein that's known to bind to different cytoskeletons elements, such as the actin or microtubules I previously talked about. And so what I wanted to know is like, what other proteins have this domain, which we know interacts with the cytoskeleton, which happens to be my passion, my interest is the cytoskeleton. And so one of these proteins that had not been previously characterized was this protein known as CG13366, which in Drosophila, that just means it's a protein that they know it's a protein. We don't know what it does, but we need to label it and we need to move on. And so I took this protein, I cloned it, and I put it into cells. I put it tagged with fluorescence protein into cells to study it. It also turned out that about the time that I was doing all of this, another group had started to publish on this protein, and they had called this protein they named this protein split disc. So CG13366 is now known as split discs. But moreover, it happened to have a very interesting story when we looked at the human homolog. So humans have a version of this protein, and it had a very interesting story. So that human homolog is called SPECC1L, which is a terrible name. Sperm antigen with coiled domains like 1, not very creative, a whole mouthful. But this protein here this is a protein diagram. We see that it's got in blue some linker domains, and then the coiled domain, which its name suggests, are featured in green. And then at the very C terminus is that CHD, or the calponin homology domain, of which I was interested in the first place. So this is what the protein looks like. And it's putatively a cytoskeletal interacting protein. Again, not super surprising, since we know that CH domains like to bind to either actin or microtubules. But it happens that there is a spectrum of craniofacial disorders, including facial clefts, wide eyes, and incomplete closure of the esophagus that happens when this protein is mutated. So there are two particular mutations, this Q4415-P, so that's basically a residue that has been mutated to proline. Gosh, I have to remember this. Now I'm on the spot. Fine. And then I will come back to that in a second because I lost it. And then a glycine 1083 to serine. And so when these two residues are mutated, in a family that has these mutations, it causes this spectrum of craniofacial disorders. And so these phenotypes suggest that SPECC1L may affect cranial-neural crest cell migration. Now, they seem like a really wide spectrum of sort of disorders, but in fact, they are all connected by this process called cranial-neural crest cell migration. I didn't go into this project wanting to study cranial-neural crest cell migration, but you kind of have to go where the science takes you. And so cranial-neural crest cells are a population of cells that delaminate or sort of pop out of the neuroepithelium and then migrate during the process of development, following these distinct patterns that eventually form the facial structure that makes up our face and our jaws. They undergo this process called EMT, or epithelial to mesenchymal transition, and they're in constant contact with one another. So when they migrate out to form these structures, they're also connected to one another. And so they emerge from a place that's going to be the hindbrain of a developing organism. And again, this only happens in mammals, invertebrates, sorry, not mammals. It only happens in vertebrates. And they migrate through a variety of environments to do this. So as they migrate, they form these structures, connective tissue in your face, cartilage, and even neurons. So these cells will migrate out and then differentiate into different types of cells. And so why these collection of mutation or collection of phenotypes are connected is because it is this specific population that is affected during this migration that leads to these craniofacial disorders that, again, seemingly look like disconnected, but are actually all connected because the way that these cells differentiate into these tissues. So when these cranial crest cells migrate, don't get to the right place at the right time, abnormalities formed in the face or your cardiovascular system and neck. So in mammal and mammalian cells, this protein SPECC1L co-localizes both with actin and microtubules. So in the top panel, we have GFP tagged SPECC1L, And we could see in the middle that tubulin, which is the other cytoskeletal element I was speaking of. And so there's a little bit of colocalization between this protein and SBCC1L. And then the bottom panel, we have an actual antibody, so we're staining for it rather than expressing a GFP tag version of this. And now we see a colocalization a little bit with actin. So this group who first characterized this protein, SBCC1L, in their hands, it looks like this protein localizes both the actin and microtubules. So let's talk about the Drosophila homologue of SB1L, which is known as split discs, SPDS. So when flies lack split discs, they don't actually all die. So it's not completely lethal to the flies. They manage to survive. But those escapers, those that do manage to live, even though they don't have this protein, they have a variety of phenotypes, including a cleft proboscis and wing blisters. Now, the cleft proboscis seemingly is analogous to what happens when humans have this mutation, but we must remember that flies don't have cranial crest cells. That's not how they develop. They develop in a different way. It just happens to be a coincidence, but it does indicate something. So here we have the fly phenotypes. The top is a wild type fly with no mutation, and the bottom is a fly that lacks split discs. And what we see are tiny little blisters on the wings. Sometimes those wings are crimpled, and then what the arrow in J is pointing to is a split sort of proboscis, a cleft proboscis, and then larger wing blisters. But what all these phenotypes together suggest is that there are aberrant adhesion in cell migration in flies. So despite that flies and humans being separated by millions of years of evolution, it seems like this protein is still really critical for adhesion and migration, even in flies. And so here we have a point where this protein is really critical to these very fundamental processes in cells. And again, why flies are a great organism to use to study this, because whatever it's doing in humans, it's still doing that in flies. And so what made me go, huh, that's funny? What really made me stop and take a moment and pause was that when I cloned this protein into the cells I study, it appeared to localize with non-muscle myosin 2, which was, again, back to like slide number 2 and 3, the whole goal of this, the whole point of this focus of this talk. So here we have GFP tagged split discs and a red tagged split disc and a green tagged non-muscle myosin 2. And so what we see is colocalization. Now, if you think back to what I just showed you looks like, what split looks like in mammalian cells, we did see that there was colocalization between actin and microtubules, not non-muscle myosin 2. I went further and did a triple stain. So we're looking at non-muscle myosin 2, split discs, and microtubules. So we're looking at all three of these cytoskeletal components. And what we really see here is a very strong colocalization between split discs and non-muscle myosin 2, and not so much microtubules. So it does look like in my hands, in fly cells, that it's a non-muscle myosin 2 associated protein. So then the question comes is like, how does this protein co-localize with non-muscle myosin 2? And so what a very classic approach in cell biology and many different types of biology actually is a structure function approach. So we're going to break down the molecule to see what portions are required for localization of non-muscle myosin 2. And to do this, we use a microscopy-based assay, and we use this, this component called Manner's overlap of coefficient. So MOC expresses the degree at which two structures spatially overlap. It takes into account the total abundance of the fluorescence that overlaps. So we're using fluorescent proteins to look at this overlap. And it results in values that range from 0 to 1. I'm telling you this because I'm about to show you a series of graphs. I want you to have some idea of what I'm talking about as I show you those graphs. So here, high values for both this M1 and M2, because you get two values, the overlap of one and the other, and the overlap of, so both two channels, the overlap of, in this case, cyan with yellow, and the overlap of yellow with cyan. We're gonna get two numbers. So high values for both those numbers would suggest co-localization. So on the left, we see two discrete structures that both the cyan protein, the theoretical cyan protein, and theoretical yellow protein localize together. And that results in high Manner's coefficient for both those numbers. Where on the right, we see that the yellow protein is everywhere in the cell, while the cyan colored protein is still in discrete spots. So will that result in a high Manders coefficient for one, but actually a low Manders coefficient for the other? Because yellow is everywhere. And again, it takes account the abundance of the protein. So yellow is everywhere, not in a discrete site. You're actually getting a low Manders coefficient because not an overlap. There's a lot of areas in that cell where there's not overlap, which is why that number is low. So that's how this, this coefficient works. And now I want to show you a series of graphs that uses that to quantify the amount of colocalization between proteins. In this case, split discs and non-muscle myosin 2. So way back in slide 3 or 4, I told you that myosin has this open or closed conformation. When it's open, it's active and bound up to actin, and when it's closed, it's kind of turned off and unable to bind to actin. And so we're able to express different types of different mutants of non-muscle myosin 2, one that is trapped in a closed conformation and one that is trapped in an open conformation. And when we do that, we can also co-express M-cherry split discs, which is in red, and so myosin is in cyan. And when we do this, we see that there seems to be reasonable colocalization. So on the left, we have our wild type situations. We have wild type non-muscle myosin 2 and our split discs in red, and we see really good colocalization as indicated by the white. This means the two proteins are in the same place at the same time. And so then in the middle panel, we have our non-phosphorylatable, or this is a trapped in a closed conformation non-muscle myosin 2. And again, we see reasonable co-localization between split discs and non-muscle myosin 2. And then in the far right, we have our open, and it's force open because of this mutation, non-muscle myosin 2. And again, we see reasonable co-localization. However, when we quantify this, so this is looking at many, many cells over many different days, we're able to quantify the amount of colocalization, again, using that coefficient of overlap. And we see that there's really no difference between the amount of non-muscle myosin 2 that overlaps with split discs. We do see a slight difference in the amount of split discs that overlaps with non-muscle myosin 2. So it seems that you see in the middle is the non-phosphorylatable or trapped in blue non-muscle myosin 2. And then in the yellow, which might be hard to see, is the open, consistently open or trapped in the open conformation non-muscle myosin 2. And this is the amount of overlap. And what we see here, what this graph is saying, is that split disc has a slight preference for the open conformation of non-muscle myosin 2. So it prefers an active open non-muscle myosin 2. So if we go back to the diagram I showed you previously, this is the diagram of split disc and not SBCC1L, but you'll notice that they're very similar in the way that they're organized as a protein. This is the diagram in the bottom left corner of the slide. And we're able to actually take out the coiled coil domains. So coiled coil domains are just helical proteins that are thought to allow proteins to coil or dimerize. And so we can delete up to the second coiled coil domain if we leave the third coiled coil domain alone. And we still see a large amount of co-localization between non-muscle myosin 2 and split discs. So it seems like those first two coils are somewhat dispensable for co-localization. And again, I'm showing you split discs in red in the far left, non-muscle myosin 2 and cyan in the middle panel, and then in the far right is the merge of those two channels, and we see reasonable co-localization. We see a lot of white, which indicates these proteins are co-localized. And this is a movie, so you can actually see the dynamics of the two proteins together. Okay. However, if we chop off that CH domain, again, that was the domain I was first interested way back in the beginning of this talk, this domain that we know binds to cytoskeletal elements. If we chop that domain off, which is what's happening in the bottom panel, on the top panel we have wild type split disc, so it has a fully intact calponin homology domain, and then the bottom panel we have our delta CH, which is the deleted CH domain. And we see that now the localization between split disc and non-muscle myosin 2 is uncoupled. They are not co-localizing. And that is drawn out here. So we can delete the first coiled coil domain or the first and second coiled coil domain and not see a huge difference in the amount of co-localization. However, if we delete that CH domain, we do see that there was a huge difference in colocalization. So that C-terminal CH domain is required for non-muscle myosin 2 localization. That's great. So we know, all right, we can delete, the first and second colloquial domain, still have that third coiled domain, but we must have that CH domain. Otherwise, we see no colocalization. So the next question we really wanted to ask was, how do these disease-causing variants affect split disc localization? Again, it is in terms of the human pathology, people who have mutations to this protein have these spatial structures that are really hard to fix and requires thousands of dollars of surgery, which not all people can afford that type of surgery. And is also detrimental to people's livelihoods. So we want to really understand how these spatial disorders come about. And so we want to see, how do these mutations affect the way this protein localizes? So on the far left, again, we have wild type split discs with our non-muscle myosin 2. In the middle, we have a Q266P mutation. This is analogous to one of the human diseases causing mutations. And then on the far right, we have our glycine 9152 serine mutation. And again, we see a reasonable amount of colocalization. And again, you know that because there's a lot of white happening here. But again, our eyes can be deceiving, and so we must quantify. And so when we quantify this, we see that there is a considerable amount of overlap between the proteins, as suggested. But if we look at the amount of split disc that overlaps with non-muscle myosin 2, or if you look at the amount of non-muscle myosin 2 that overlaps with split disc on our left panel here, we actually see a little bit increase with the amount of the Q266P that overlaps with non-muscle myosin 2. And then we see a decrease with that G915S mutation. When we look at the inverse, the amount of split disc that overlap non-muscle myosin 2, there's a little bit a decrease for both situations. But what this suggests is that non-muscle myosin 2 has a slight preference for that Q266P mutation. Again, it's not a huge difference, but when we're talking about molecular details, particularly when we're talking about the migration of cells that need to go from point A to point B, everything has to be at the right place at the right time. Any deviation from that can cause and lead to huge problems. So the fact that we see a slight preference for this mutation might be an indicator that there's something going on with this mutation in terms of cell migration and in terms of localization of this protein with non-muscle myosin 2. Okay. So we also wanted to look at the CH domain alone. So again, I chopped that off previously, and we saw there was no co-localization with non-muscle myosin 2, or very little co-localization non-muscle myosin 2 with split disc when we delete that CH domain. So what if we just express just that CH domain alone? And naively, I thought this was it. This is the key to how this protein localizes non-muscle myosin 2. But as you can see, hopefully from these three images, that when we express just the CH domain of split discs alone, we do not see co-localization with non-muscle myosin 2. And so again, on the left, we have a red tagged CH domain of split discs, and it's everywhere in the cell. It's not discrete. And then we have, again, non-muscle myosin 2 in the middle panel, and then merge of the two. And so it's really critical that CH domain, it does not actually localize by itself, which actually suggests that dimerization is required. So that's also not too surprising, even though I thought this experiment would totally work out. It's actually not too surprising because, again, this molecule has a ton of these coiled domains. And as I alluded to before, these coiled domains allow the protein to wrap around together. And actually, what we think is that two individual molecules of split discs wrap around each other together to form a complex. What have I told you so far about the co-localization analysis is that split discs may have a slight preference for the open conformation of non-muscle myosin 2. And then we're following this up with pharmacological manipulations. So it's great to show this sort of open-closed controversy with both the mutations I showed you with and also with drugs that inhibit non-muscle myosin 2. And that deletion of the CH domain appears to uncouple split disc of non-muscle myosin 2. So when we cut off that C-terminal CH domain, we no longer see co-localization. And then also, if we look at the mutations, it looks like that G915 mutation appears to decrease the amount of co-localization, while that Q266P mutation appears to enhance that co-localization. And then if we express calpone demolimate alone, it does not localize with non-muscle-myosin 2. So while it's important for us to understand how split disc is localizing to non-muscle-myosin 2, what's really critical here is understanding the process of cell migration. So what we're looking at here is cranial neural crest cells migrating from the hindbrain during development in a chick embryo. And so in green are these neural crest cells as they migrate out from their place of origin. Eventually these cells would go down and make the facial structures for the chick. And so again, they must migrate to form these structures, and that this pathology that's in humans is caused by these point mutations rather than deletions. So we really want to understand how the point mutations are affecting migration. And so a larger picture that we're trying to understand is what is the role of non-muscle myosin 2 during cell migration? And then within that is how does this protein split disc affect that? So cells make attached to the extracellular environment through these receptors that are coupled to these protein complexes called focal adhesions. And here we have an example of focal adhesions in a Drosophila cell. These are basically the grips or the tire treads that a cell makes with the external environment as it's trying to migrate through or around a substrate. And so we can study these dynamics of these adhesions to get an idea about what's happening in terms of adhesion and migration. So our hypothesis then is that split disc plays a role in non-muscle myosin 2 regulation. It's possible that focal adhesions are also affected. And so what I had my student do was basically measure the dynamics of these formation of these complexes. As they form and as they disappear, that gives us an idea of how the cell is migrating and adhering at the same time. And so when we got rid of split discs, we saw no difference between the assembly rates of these focal adhesions. However, if we look at the disassembly or the undoing of these structures, which is equally important as the formation of these structures, right? If you bind to something as you migrate along, you don't let go, you're going to be held back by the fact that you can't let go of this substrate. You can't let go of the past, if you will. So being able to assemble and disassemble is really critical. So what we find is if we deplete split discs by RNAi, so we deplete the amount of protein in the cell, we actually see an increase in the rate of focal adhesion disassembly. And again, it's a very subtle difference, but it yet is a difference that exists. And again, I just want to point out that it's super critical that cells get to the right place at the right time to development. If they don't do that, these structures don't form. So even subtle differences can have a huge difference in terms of how the organ plays out in the formation of facial structures. But again, the disorders that we see in people, the pathology that we see in people aren't caused by deletions of this protein, they're caused by these point mutations. So again, we wanted to look at that. And so when we look at the Q26P, Q is glutamine, by the way. I had a minor senior moment and could not remember it. This Q26C to P mutation, or this G915 to S mutation, we look at the formation of those focal adhesions, we see no difference. So again, the assembly is not affected by these mutations. But what we see is a huge difference in the disassembly rate when we express that Q266P mutation. So it seems that that mutation somehow is causing these focal adhesion disassembly rates to increase almost 1.75 fold, almost 2.0 fold increase in the disassembly rate. So this is interesting and very important because it shows that there's something going on with adhesions that's not quite right with these cells. And that could possibly lead to the pathologies that we see in humans. I'm not going to say it does. This is a possibility. But this is an interesting aspect of what's happening with this protein. So what I've shared with you today is that split discs in the human homolog SBACC1L co-localizes with non-muscle myosin 2, and that CH domain, that calpone homology domain, is needed for this co-localization. Depletion of split disc increases the rate of focal adhesion disassembly, and further expression of that glutamine 266 to proline variant further increases focal adhesion disassembly. I want to thank my crew. This picture was taken last fall before COVID hit, and we were able to get together and enjoy some food together. But some of the people that worked on this project are in this picture. Again, I'm not sure if you're aware, but Reed is a small liberal arts school. It's undergrad only. And so my research team is a bunch of very talented, determined undergrads. My funding, National Science Foundation, National Institutes of Health, and Reed College itself. My collaborators, Peter Bar Gillespie and his group at OHSU just across the river, and Greta Glover, who is Dr. Greta Glover, who is a instrumentalist in the department, who helped train some of the physicists who are a portion of this project I wasn't able to share with you today. And I will, now I will transition into my sort of personal story. Again, this is more of an aspirational goal, something I work for, which is also wrapped around who I am as a scientist and where I'd like to be as a scientist. And the question that I think about a lot in terms of teaching, in terms of research, is what can we do to make sure that everyone gets a seat at the table? Because science traditionally has been a barrier for everyone to come and feel like they can be welcome and be a scientist. We're all born scientists, and then something happens to us and some of us feel we can't go back. And some of that is systematic, right? The country has a lot of problems with systematic racism, and it has percolated down into science. And in fact, science has been used, in fact, to perpetuate racism. And so I really want to work aspirational to make it a place for everyone to come and have a seat at the table, and that we all are capable of being scientists. So how do I do that? One is I bring embodied diversity. I am a Black gay man, and I bring that embodied diversity with me to the classroom, to the research lab, everywhere I go. And I think that's important. As an undergraduate and graduate student, and even as a postdoc, I never had a faculty member that embodied the type of diversity that I had. Never had a Black faculty member, never had an out faculty member that I worked with, that supervised me, or that I took a class from. And so bringing that embodied diversity to my students is really important to me. The other thing that I try to do is to tell my story and erase the facade. So a lot of times students think, oh, professors are some sort of godlike being that are all-knowing and all-telling. And the fact of the matter is, it's not true. We make mistakes. We don't know what we're talking about a large percent of the time. And I think that they should know who we are as people, and that we are people. And so I try to erase this facade and break down these barriers so that they realize, oh, this person is just like me. And if they can do it, I can do it, which is exactly the point I'm trying to make. So a little bit about my story. I was born in Brighton, Colorado, which is a small town outside of Denver. And I was a 4-H kid, believe it or not. And so what that meant is I raised rabbits, and I also had horses. And so this was some of the earliest influences on my scientific career, because I was surrounded by biology all around me. Not only the farm animals that we had, but the sort of the country setting where I could pick up toads and see snakes and see birds and have all this wildlife around me really influenced me and gave me a passion for science. I also was a pretty serious athlete in high school. And so these two loves sort of conflicted with one another, because it's hard to be a 4-H kid and also an athlete. And that as I got older, sort of the athlete part of me won out. And I eventually earned a scholarship at the University of Michigan, where I went there on a track scholarship. And I ran track at Michigan. It's a great institution. Really enjoyed my undergrad education. Large part of it was being part of this team, this track team that was the reason why I was there. From there, after graduating, I went to Northwestern, just across on the other side of the lake, if you will, for grad school. And there I ended up working for a man named Avery Posey, who was world-renowned for his understanding of both actin and microtubules, and I studied those two projects in his lab. This is one of, if not the first, but one of the first images I ever took in his lab way back in 2000, I don't want to mention. And what we're watching is a cell migrating. And at the time, I didn't even know cells can move. And so I was fascinated by this process because I still don't understand what's going on here. And I really want to, that's what drives me, trying to understand what's happening here. Excuse me. After graduating from Gary's lab, I moved on to my postdoc at the University of North Carolina, Chapel Hill. Go Tar Heels, I guess. Where I worked with Steve Rogers. And Steve was known for his work in S2 cells, these fly cells, and RNAi, which they're very sensitive to RNAi. So it allows you to actually deplete cells and work with cells with RNAi depletion. And that's where I picked up the system that I'm currently using now. But I just want to stress that postdoc was really, really hard, particularly being a person of color, minoritized individual in science. It was really, really difficult. And I think the only way that I got through was finding people that I could identify with and who identified with me. And one of them was Omar Quintero, who's now associate professor of biology at the University of Richmond. Omar was one of those type of people that would send me his grants, would read my grants, would tell me what he was doing and suggesting that I should do the same thing. So he kind of reached back and lifted me up. And if it wasn't for this relationship with Omar, I am definitely sure I would not be from where I am for now. And I cannot thank him enough for being a mentor and being a colleague and just being someone who really looked out for me as I was coming up as a graduate student and as a postdoc. From UNC Chapel Hill, I got a position at Reed College, which again is in Portland, Oregon. And I was able to bring this fly cell system to undergrads. And it turned out to be a great sort of marriage. Fly cells are pretty robust. They can take a licking and keep on going. And it turns out that's great for undergrads who are learning how to do tissue culture and they're new learners to cell biology in general. So it ended up being a really great sort of system for students to learn. So again, back to this idea, this aspiration, how to make sure everyone gets a seat at the table. I think it's really, really important that people recognize that you're biased and that you work in ways to counteract that. You know, even though I am Black and I am part of the LGBTQ+ community, that doesn't mean that I don't have biases. And so recognizing that I have these weaknesses and trying to counteract them is super important. The other thing that you must do if you're trying to make sure that you're making an inclusive space for everyone to come is to try to meet students where they are. Not everybody has the same preparation. Not everybody's had the same experiences. Not everybody's had the same resources. And so trying to understand that and then meeting students where they are and showing them that they can still achieve no matter where they started from is really, really important. So I try to keep that in mind too, as I'm interacting with students and as I'm teaching students, mentoring students, is trying to meet them where they are and trying to find things, trying to find ways that we can make this work together. And then what I have been able to do post-tenure, pre-tenure a little bit and post-tenure now, and that's how these things kind of work, is that I've been able to actually start influencing the broader STEM community and through advocacy and work. And so I'm a member of, I'm the co-chair of the LGBT Task Force, which will be an actual official committee for the American Society of Cell Biology, which is the largest society of cell biology in the United States, and also is international because it's part of EMBO as well. And so my role there is to guide this committee in advocacy for the LGBT community within the society, and I really enjoy that role. I'm also part of the Equity, Inclusion, and Diversity Committee of the Genetic Society of America, again, where I am sort of advocating for minoritized individuals within that society. And this is really important to me as well because, you know, I had a postdoc way back in the day tell me to just stick to the science and everything else will work itself out. And the fact of the matter is that's not true. I mean, we are humans, and as humans, you know, science isn't a human endeavor. And so we humans bring our humanness to science. And that means the bias, that means the systematic racism, that means all the things that, the ugly things and the good things that humans bring to whatever they touch, but also they bring it to science. Science isn't just facts and measurements. It's more than that. And so we have to recognize that and realize that science is a human endeavor, and then work with that, knowing that it's a human endeavor, and work to change the culture as such. So this is my journey, if you will. And I again want to thank the organizer for this opportunity. And anytime I can get out and talk about my students, about Reed College, and about the work we're doing, I feel very fortunate for that. So thank you for this opportunity, and I will entertain your questions.
All right, everybody. Thank you to all of our speakers for showcasing your talents and impactful science, and to everyone who is watching. Tune in same time, same place at blackqueertownhall.org. See you tomorrow. Dr. Brady, that was an amazing